formula is going to be defined by the have in the metadata, and you can use multiple things that are important, and the PCA is the one who is going to give us that information, for example, right? This is clear, I think, so we are good on this. And maybe this is the only thing you need in your project. There's nothing else you may need. Okay, we talk about results. I don't think I need to review this. Any questions about this? No, right? This is the output that you get when you do your analysis. It will tell you the comparison on top, right? We also talk how you can, if you have multiple levels in your column condition, you can do pairwise comparisons or access any comparison you want using the contrast argument in your results. We did that. That was one of the last exercises we did. And even if you have multiple treatments, it doesn't matter what you use as a baseline at that point, right? You can call it however you want, but you can still do any pairwise comparison using the contrast. It doesn't change. Internally, you know, DSeq2 does the same operations. There's no difference. I said I probably didn't explain that again, but internally, this is how it's happening, right? It calculates a baseline and uses another sample to estimate some fold change, and does the statistical analysis. But okay, so here is where it got a little bit more complicated, right? This was the example of yesterday when we were talking about the PCA and how you can have multiple sources of variability in your data and can affect or can inform you how to create a more complex model, right? When you want to model the strain or the gender and the condition under study. So you can build a more complex model. So DSeq2 is going to calculate multiple coefficients. And batch effects are not different, right? So you can have a batch effect and you want to model it. And I put this example where you have your experiment. You notice there's a lot of noise in your data. But you know that it was produced in 2 different labs. And you notice the baseline for both labs is different when you plot them separately. So you know there's some sort of batch effect. And you can model it in your formula. If I'm going too fast now, I mean, we did this yesterday. I'm just reviewing it just to be sure. OK, so this is what I was saying, right? So you can add it in, because it's a column in your metadata, and you have information what lab did what. You can include it in your design formula, and DSeq2 is going to try to model that effect, the effect of the batch. So you are not removing it, you're just modeling it. So it will calculate the coefficient for the batch. And I was explaining this is how internally it's going to be done. You have a baseline. It's going to calculate the batch, produce a batch slab B, and then it would estimate the actual fold change that you want to calculate. OK, here is where I was trying to say, let's forget about all these matrices and everything, how internally it's happening. And I was trying to verbalize what you're doing in your formula, right? So what we're doing here is saying, because by default, so this is important, by default, DSeq2 is only returning one table, one coefficient, right? One fold change. Well, one fold change for every gene, but only one table, which is the last element in your formula. If you swap it, the last element in your formula will be batch, and when it returns by default only one, it would be the coefficient for the batch. You don't have to change the design formula to get the batch. Internally, you can use the contrast again, in the same way we did it, and obtain whatever effect is the batch. But by default, that's the idea, by default, this is what it's returning. The fold change is caused by the condition, whatever levels you have in there. And why I'm saying that, because again, you don't need to know how things happen internally. You can phrase this as, OK, regardless of the batch effect, because I'm not going to get that reported at the end, I'm calculating the effect of condition. That's a way that I have to phrase it. I don't know, you can phrase it probably different. You could even say, oh, removing batch, I'm just getting the condition effect, for example. But again, internally, coefficients are calculated for both. It doesn't matter. So forget about this. So this is the question I was asking yesterday, right? So if you want to know the effect of a sleep deprived regardless of the genotype, so the effect of condition, and we don't care about the genotype, so this is how you would do it, right? Because we know that the last element in our formula is returned by default. Yep. So now, yesterday I added this slide. Is there any difference here? Sorry, what?
The second one, the genotype will be the...
What it returns by default. But internally, this is identical. There's no difference. Zero difference. So, but by default, there's the difference that we're talking about. It will return a different table. If you don't tell him, give me a contrast of this, this, and this, DSIC2 by default knows this is the last element in your formula. It will return that coefficient. But internally, everything is calculated in the same way. There's no difference. Right? Only the default output. So when you do results, DDS, alpha, whatever, return me this, it will return a table using the levels, in the first case, whatever is in condition. And in the second case, whatever is in the genotype. But if you use the contrast that we practice, you can obtain the other element in your formula. So if you don't want to think about changing returns, the only thing you need to do if you want is repeat everything and changing the elements in your formula. That would be the easiest way for you mentally, right? So you don't have to know exactly what is happening internally. So, but there's another thing. I think we are clear on this because I wanted to clarify this, right? So when I'm phrasing it in the way that I was phrasing it is like, okay, ignore genotype, give me only the condition. You would use the formula on the top. If you want the other way around, say, okay, remove the condition effect, quote, quote, we are not removing it, but you remove it, and just return me genotype, you would do this and do a normal analysis and don't use contrast, right? So, but we can have situations like this, right? So this is one gene. We have the wild type, and we observe that there is a decrease for that gene. But when we change to SHANK3, right, the knockout, we see that the behavior is completely different. It goes up. So this is quite unexpected. It can happen, right? So we have what we call an interaction. So the effect of having a knockout changes the behavior of that gene. And this is what generally we call an interaction. So a sleep deprived interacts with the genotype, and we get a completely different effect. Can you figure out what is the interaction effect? Yes. So now we add another element into our formula, right? You can add what we call the interaction effect. Is expressed in this way, which is combining both columns in your metadata. It doesn't matter the order. You can say condition genotype or genotype condition. Doesn't care. It's the same expression. And you are calculating the effect of the interaction term, which is really clear here, right? One is going down and the other goes up. But if you have something more subtle, right, one that probably is a flat line, and then all of a sudden you get a little bit of increase, you want to know, is that little bit of increase significant or not? So then is when you would want to know if there's some sort of interaction effect in your design, in your experiment. Questions about that interaction effect? I don't know how often you're going to use this, but I have the feeling that, oops, sorry. Keep pointing to the screen, but there's nothing there. I know it's early in the morning. Stop me if you have questions, please. I forgot what I was about to say. Questions here? Did I explain the interaction effect here, what it means?
In this equation, if in the last that this mustard thing, that genotype colon condition.
Oh, this one here?
Formula. If I write condition first and genotype later on,
It's the same.
it's the same.
It's the same. Oh yeah, I was explaining why I think is important this type of formulation in this example, because you probably want to study what is the interaction of knocking out something when you sleep deprive it. You probably don't care. So let's say yesterday we observed how it was called PLIN4. It was called that gene, the gene that we saw big differences. That gene changes in both situations in the same way, right? So why to work with the genotype? That gene is not important for the genotype, right? It has exactly the same effect. It doesn't matter if it's normal or if it's genotype. Maybe you care about the genes that behave differently when you have a knockout, right? That interaction of not sleeping and your knockout are probably the interesting genes for you. I have the feeling the questions that we were asking yesterday is these type of questions. Does the condition together with the genotype produces a different effect? I think is what the paper is trying to answer. Yeah, I think, did I explain that more or less okay? So I don't think the paper cares about PLIN4 because it's a gene that changes in the wild type already and it's changing also in the genotype. Okay, sure. I mean, it could be interesting to know. So we know that PLIN4 is really affected by being sleep deprived. That's it. Not by the knockout. Because the effect looked like pretty similar to each other. There were no difference in effects. But if we have this situation, this is interesting. So when you are sleep deprived, but you are, let's say, a normal mouse, yeah, it goes down. But all of a sudden, they knock out a gene, and then it goes up. So this is an interesting interaction between being knocked out and sleep deprived. So I think this is the type of genes that the paper is trying to find. Again, this is clearly, I mean, you don't have to do an analysis. If you plot these, you're going to see there is an interaction term. But an interaction term could be also, it goes up for wild type, but it goes even higher up for SHANK3. Then it's hard to evaluate if you don't have a way to model it. There is an issue of this interaction term, and because all this is linear, right? So you have a fold change of 2 going up in the first condition wild type, and you have a fold change of 2.5. Is that enough for the linear model to detect the signal? Do you want me to go through this idea? No? Okay, there's a no here. But you got the idea, right? But if you want, you could do the whole exercise, calculate the effect of the condition, the genotype, and this thing here. So it's the same example. It goes down and it goes up, right? So the fold change you're going to detect with the interaction is this orange line. Because the linear model is expecting, right, it goes down for the wild type. The linear model is expecting that it would go down for the knockout. So your knockout should be here, right? Because this is the condition effect. So the knockdown should be here. But all of a sudden, we found the knockout is up there. So the fold change you're going to get is this orange line when you calculate the interaction term. And again, the same idea. If you put it at the end of your formula, by default, DSIC2 calculates the interaction term if you don't ask for anything else. Sorry. Oh, yeah. So I'm not going to go through this. We said that we are not going to review any more of this. But yeah, you can again, calculate other things that by default they are not calculated. The purple line here, which is how it changes in SHANK3, in the way we formulate it, it's never calculated by DSIC2, but you can insist to us using contrast to calculate this purple line. All this is part of the DSIC2 vignette. If you want to read more about it, you can go there, and I think it's explained more or less. It's not in too much detail, but it's explained. So same situation. I'm going to give another example about the interaction term. You can imagine you have this type of situations where you have a control, you have treatment 1 and treatment 2, and you have the combination of both treatments. This is probably even more clear than before. You want to know what is the interaction. So the linear model is expecting, okay, it's going up when I do treatment 1, it's going up when I do treatment 2. So the expectation is you're going to add treatment 1 and treatment 2 together, then the fold change for the interaction should be that, the addition of treatment 1 and addition of treatment 2. But what if there is an interaction, right? What if instead of adding both fold changes, all of a sudden you get 10 times more? That implies there is an interaction. Again, based on the linearity of the calculations we are doing. So everything is expected to be linear. It doesn't mean biology is linear, though, right? But it's exactly the same situation. You would do treatment 1, treatment 2, and then do the combination of both. And you are calculating the interaction and figure out if. So.
And it could also be temporally that what you treat first, treatment 2 plus treatment 1, or like.
Okay, if you are a temporal thing in there, or that's a bit more complex, because you're going to have to.
Is it the same if you do treatment 1 plus treatment 2, or you do treatment 1 first and then treatment 2, or is it the same? Or if you do treatment 2 first and then treatment 1?
Well, here I'm assuming you do it at once. Assuming it's a drug, I'm calling it a drug, and you give the drug at the same time. If you add temporary effects, you need to, this is going to be more complex because you need to test the timing between drugs, I guess, and however you do it. I call it drug, but it could be, as you say, any treatment, yeah. So this is what I was trying to, forget about the metrics for now, but these are the effects, the individual effects of both of them, right? So if we add them, this is the expectation. By linearity, if you do treatment 1 and treatment 2, you're expecting that the combination of both, your data should fit here. But you realize, no, it doesn't. We have some extra thing that is not expected. So this is the interaction, or the synergistic effects, or however you want to call them, right? So this is the interaction term that DSIC2 is calculating, and basically is telling you, is this different than 0 or not? So that's the p-value that you're getting. Yes, so I think this is the example before, right? We said that once you do your PCA, you can observe if your expectation of the condition being the main source of variability of your data or not. So you can build more complex models if you want to. But remember, by default, if you put it at the end, that's what DSIC2 returns. So you care about the treatment, but you don't care about the other two effects. So you try to remove those effects. You will put it at the end. OK, so we've seen 3 things that we could use in our example of yesterday, the Shang3 and the sleep-deprived. We could have a single condition, like we did, how we call it, the condition geno, whatever we call that column, where we combine both. Yep, yep, so that. So we can combine both. We can have 4 levels, and we could do this analysis. And then you would need to do pairwise comparisons and try to understand the difference between those and what it means, right? We could remove, for example, again, it's not removing. We can model the genotype effect and only care about the condition. Or we can add the interaction term, which is what I think the paper is trying to do, trying to figure out if having a knockout has a specific effect on the sleep deprivation, or what genes have some sort of interaction term with the knockout and the condition. Yes? Did I do a better job today than yesterday? I think I did, I hope. Questions? I mean, again, it could be that you only ever use DSIC2 for the first example, but you can do this and even more complex models that don't follow necessarily the linear model. You can have, if you know that, this is a bit more complex, but we can talk about concentrations of a drug. If you give 0.5 and you give 1, or you give 2, or you give 3, or those concentrations, if you go basic like this, you are expecting that if you do 1 milliliter of a drug, or you do 2, the linear model is expecting double effect. But we know that it may not be true. If you get double of a drug, all of a sudden you get a big effect. So then it gets more complex, right? Then it's not linear, and you need to build some sort of curve or assume how many. You're going to have to assume some things in your model about how the drug, or if you know it, if you've done some experiments and you know it's not linear, then you need to build a different models. In those cases, your friend is you need to find a friend that is an statistician to tell you how to build these models. Or I recommend you to talk to one, right? Oh, okay, I stop here. I thought I would have more complex models. You can do also, another thing that sometimes I encounter is, what do you call them? When you do different time points. Temporal, yeah, it has a name, right? Well, it doesn't matter. Let's imagine you have time points, right? There is a tendency of do pairwise comparisons, comparing time 1 with time 2, time 1 with time 3, time 1 with time 4, and so on, and so on, and so on, right? There's ways to do a single model and test if your gene changes across time or not. If your question is, if your gene changes across time, you can build a simple model, some model that just tests that, if changes or not. If you care about the comparisons, yeah, then you should build a model that you can compare each time point. But it can get even more complex. You could add a genotype and say, does my gene changes depending on the genotype? Then your model gets even more complex. But you can do even temporal or time scales or time points. I forgot the name now. But yeah, you got the idea, right? So you can even go more complex. I think that in the vignette, they may give one example of how you can build a model with using time points. The statistics changes a lot when you do that. Because you can build two different models. You can even test. I have two models, right? Simple model. I mean, we can do this, I guess, as an example. You can build two models, and these two can test if this fits your data better than this or the other way around. You can even test that if you want to. Again, we are not going to go through this, but you can build two models and test, okay, is this model fits the data better than this model per gene? Okay, questions? Is that any of you doing this bulk RNA-seq analysis or planning to do one? Oh, okay. Oh, you guys. Okay, well, if there are questions, we can talk about how to build models later if you want to. No, you want me to? I was about to wait, but Yep, yeah. I'll switch it up because I saw people working in it, and the day before there were people working in it too. And I noticed that I terminated and somebody was still working maybe. So I'm sorry if I interrupted an analysis of somebody.
Is the AWS like for a student? Is it discounted
Cheaper?
amount or
So if you want to try AWS for whatever reason, when you sign in, you get $100 the first month. So you can use $100 for whatever you want the first month. After that, you're going to be charged by a credit card. There are two types of accounts, the basic one and a one that allows you to do more things kind of thing. But the basic, when you sign in, they give you $100 and you can use them for whatever you want.
Yeah, I think for the type of exercises we do, like in this course, you should just run it locally. Like I downloaded all the notebooks, and I think you can, if you just install like R.
Oh yeah.
You should just run it.
Yeah, no, it's totally fine. Everything we run, with the files that we run, or even more files or whatever, more samples, you can run them in a laptop, yes. The single cell analysis we're going to do today, it's not working, right? So give me a second. Come on. Cool. Thanks for answering. Yeah. So, yes, pretty much everything we do, you could run in a laptop, correct. Today, depending on for the single cell RNA-seq, a laptop can be quite limiting depending on the number of cells you're working with. We had another question about AWS. There was other questions about AWS? Besides that, oh, students. Yeah, you mentioned students. Yes. I don't know if there's any other. I know you can ask, and I actually did, but too late. You can ask money for education. Like, oh, I have a project, or research projects too. If you have a research project where you need compute power, you can fill a form and request money. Up to.
So they will give you a discount on that?
No, they will give you credit. It will give you $5,000 for doing research on single cell for something. You can request that. So like they will give you an award if they consider that your research is important. Or if you have a workshop like this. Yeah, but we needed to be, you need at least 90 days in advance. And I did it, but it was too late. I don't know, we may get something. So yes, so if you have research and you want to use AWS, you can request money. You write a project, and if they consider the project is interesting, they will give you credit.
So they will just give you credit hours to use the server?
Yep. And they're going to ask you, so you're going to have to justify it in the sense, oh, I need this much computer power.
Then when you write something, you have to acknowledge them.
Sorry, what?
Then you'll have to acknowledge AWS that this is
Yep. Yep. And you need to be specific about how you're estimating the cost, right? So if you're saying, oh, I need this, this computer power, I'm going to use the Mumbai server, and I know it's going to cost me X, and I want that money, kind of thing. Cooking is winning. Okay. Sleeping second. Is that a single answer? Guitar, novels, photography, and traveling? Is that a single answer? Oh, that's cool.
Single answer.
Yeah.
Somebody, not me.
All right, a friend of mine, right? Yeah, sure. Rifle shooting. There's shooting ranges around here? What about, is hunting something that people do around here? Go hunting? No? And so I'm not a hunter. I'm not going to ever hunt, and I'm not a hunter. I used to fish when I was a kid, but not even anymore. The what? A cowboy? I don't know if it look good to me. In the area that I am, I like running on the trails. And there's signs, of course, about hunting, right? I'm always afraid that somebody is going to shoot me one day when I'm running. But the cool thing is most of the forests, the only thing they allow hunting is with archery hunting, like with, bow and arrow. That's the only hunting they allow in my area. I don't know who does it. I've never seen anybody with a bow and arrow around there, but it's deer. I'm assuming they hunt deer over there, but I don't know. Well, cooking is winning. There's a lot of cookers here, I guess. Cooks. No cookers, cooks. Even my English is going down. Okay, today's topic, single-cell RNA-seq. How many people plans or is doing RNA-seq or wants to do a single-cell RNA-seq? Okay, we have 4 or 5. Okay, a few. Good, good. So the reason when people comes to me and say, oh, I want to do, or why would you don't organize a single-cell RNA-seq workshop? And I say, yes, I do it if we talk about bulk RNA-seq first. If we don't talk about bulk RNA-seq, I'm not doing it, kind of thing, right? So I think talking about bulk first, it gives you an idea of the things. There are enough similarities about the process between them that is worth talking about bulk, which is a bit simpler than just jumping in directly to single-cell RNA-seq. forget about the question. So that's what we've been saying these days, right? So when we do bulk, we lose some of the diversity that is inside of that tissue that we are analyzing, right? Not all cells are equal. Not all cell states are equal. And there's interaction between the cells that we may want to study. So that's why we use single cell when we care about those questions. Bulk is still a worth type of analysis, right? I'm not saying it's not worth doing it. But if you care about this type of questions regarding about cell type, states, and interactions, maybe you need to consider single-cell RNA-seq. So this is a situations that when I was doing the slide came to my mind, but probably there are others, right? So if you want to identify cell types that are present in your tissue, or if you want to identify some sort of unknown or rare cell type, so bulk, this is not going to be able to do. Of course, if you care about where a thing, Dr. Amit was talking about this yesterday, right? Lineage tracing, you don't have to use single cell. If you want to just work with one cell type specifically, if you don't do sorting somehow, you're going to have to use single cell RNA-seq. I don't know if we talk about this. This is also probably a little bit more recent, but there is, you can do analysis for communication between cell types. If you care about how cell types communicate, there's ways to do that in single cell RNA-seq. Any other applications that you can think about? I don't know. This is the ones that I'm thinking the most, or the ones that I've seen. So Rick Pierce yesterday, he clearly cared about one cell type, endothelial cells. I don't know if you remember, that was the thing, the project number 2, where they put the wire on sick kids. They get the wire, and the endothelial cells were a tiny, tiny fraction that there's no way to recover that if you don't do single cell. We've seen this before, so I add it. I don't want to be part of the club that doesn't have the fruit part of it. So I'm part of the club now. So we understand those differences, right? So if you care about the elements, of course, you need to do single cell. This is the slide that I have for a similar question to compare about bulk or not. So, and the message we've been trying to also to impose here for a lot of things. For why do I just check the top 10 lines of a FASTQ? Why I'm checking every time I'm printing a new object in R, or why I'm plotting it? Okay, so you always want to visualize. We recommend to visualize what you do. Check what you're doing, right? And normally implies that some visualizations. So this is not single cell data, right? This comes from something else. But I saw it, actually, I took this from, I don't remember where I took it from, but somebody else used it also to talk about single cell. And I thought, oh, this is a cool idea if you're talking about single cell to show it, right? So this is data where the average expression, the bulk expression, if you want to call it like this, is the same. But internally, it could be plotted in different ways, right? This could be cells that they are clustering completely differently, right? And you're missing this information. If you do bulk, you would miss this. You just get this number. If you do single cell, then you realize how they interact between them or how they cluster together. Or if you get a dinosaur, you don't get a dinosaur.
What do you mean by this? Visualize your data.
Everything. From the getting a plot, doing a PCA, exploration data analysis, we've done a lot. And MA plots, I don't know. Any plot we do is visualizing your data, right?
All
Or
analysis that have been doing gradually, that
yep, that's visualizing the data.
metadata, right?
Yeah, overlaying the metadata and PCA, all those visualizations. If you don't do it, you're missing that piece of information, right? Or even just printing the object that you are building, right? Double check. That gives you an idea what is happening internally. It's always good to visualize. I'm putting the message beyond just a plot. I'm saying visualize everything you do is always helpful.
Even when you
Yeah.
Sometimes you have to go back to the lab, look at
Yeah.
the PPT, and
Tell me about it. So we can talk about that tomorrow, but visualizing alignments or variants, we are not going to do it for the sake of time, but yeah, visualizing alignments, it's really important. Or I'm hoping that one day we don't need it, kind of thing, or we need it less and less, but when we first working on it, we used to do it constantly all the time. And clinical settings even more important, right? Because you need to be sure that what you're detecting is real and it's not an artifact, definitely. It's always hard to compare type of technologies when they're so different, but definitely it's one of those have their own advantages and disadvantages, right? We can say that bulk is easy to analyze. You don't need the same computing power that you need for single cell. It's more, statistically, it's more robust probably. There are models that are going to be work better. Single cell RNA-seq is still evolving. There's still new statistics, new programs. They constantly get updated. You need to, if you want to be in the cutting edge, you're constantly updating the software to the latest one, right? So each one of them has an advantage. Cost is one of them, right? And their own applications. We already talked about this, right, in the first slide. They may have different applications. So I'm not going to say that single-cell RNA-seq is always the answer. That's what my point, right? So depending on what you're trying to do, bulk RNA-seq is probably also an answer. And for whatever reason, because single-cell is on the hype, right? You're probably going to get a better publication if you do single-cell RNA-seq, or that's the expectation sometimes. I've seen cases where people sort the cells first, and then they do single-cell RNA-seq analysis. What is the point of sorting in the first place, right? Don't sort if you can. And probably, I don't know. Sometimes I'm a little bit shocked what people do. And sorting is going to affect the expression. You are manipulating the cells even one step more than you need to. It's going to change the expression. Some people forget about some of the things I'm putting in this slide. You probably know them. And we talk, at some point, I think Bonnie mentioned them, right? So there is one problem. The low depth of sequence we're using, right? So you're going to see that our count matrix yesterday had a lot of numbers. We have zeros, for sure. But it had, you have expression in a lot of genes. You are able to detect thousands, 20,000, 30,000 genes in your data.
And also in sequencing, you're wasting a lot of sequencing actually. I mean, like, what are you sequencing? I mean, like, there are so many barcodes.
Yeah.
The actual sequence might be.
Yes. I mean, generally we don't care about the actual sequence, but it's true. I mean, if you could do shorter sequences, maybe it would be cheaper, the analysis. You're going to get a lot of zeros. The problem is a zero in your cell, it doesn't mean it's not expressed. It could mean that it's not detected. Right? Actually, I saw somebody was doing a statistical analysis. We always say, oh, your data is zero inflated because there's technical issues with single-cell RNA-seq. So somebody did a statistical analysis, and it seems that it's not necessarily true. The sequencing we're doing for single cell is already enough to tell you that you're going to get zeros. It's going to be impossible to do enough sequencing to cover all the genes. So it may not be necessarily technical, the presence of zeros, considering sequencing non-technical. Right? We always say that, oh, the whole process of library preparation, using the microfluids and everything, at the end of the day, you are not going to be able to detect all genes. It seems that if you look at the numbers, somebody did some calculations, the sequencing they're currently doing for single cell, it seems enough to explain why we see so many zeros. But forget about it trying to sequence deep enough that you're going to detect all of them.
So I actually want to add one point on that. So there is an idea called as sparsity. So it's actually relevant for both sparsity, data sparsity, and actually this is relevant for single cell as well as for the spatial transcriptomics. So the question that you should think about asking that what is the possible number of genes that are expressed in a given cell? Just to take one random cell and ask like how many genes might be actually, you really need actually a little more number of genes to be detected. And one of the way people are trying to do is to bumping up the sequencing. And again, what Francis is saying is right, that it's hard to capture all the cells, all the genes that are actually expressed in a single cell, because that means you are actually going to sequence for an indefinite depth, and nobody knows that. But nowadays, because the sequencing costs are actually going down, people are going to sequence almost 5-fold more sequencing than the recommended sequencing depth. So like for 10,000 cells, they are sequencing a billion reads, which means you are sequencing approximately 100,000 reads per cell. And that actually allows you to see more number of genes per cell. So, but again, the whole idea of sparsity, that sparse distribution of genes, which is way beyond the number of genes that are really expressed in both the data sets, single cell as well as in the spatial data set, exists. And this is a problem, and that is why a lot of people consider single cell as a qualitative data rather than quantitative. And the statistics and bioinformaticians says that the other way of looking at it, that the way they are doing differential gene expression or quantitative expression in single cell or spatial is through bulking. So they have a cluster and then bulk everything together, and then they are doing that. So that gives them the statistical power to define this as a statistic. It's a quantitative data. But again, this is a point of argument, and there's another point of argument about it, which Francis will touch, is about replicate. So the problem with single cell, a lot of people consider single cell as if you're sampling 10,000 cells, that means it's a 10,000 experiment. That's why they say we don't need replicate. But without biological replicate, nothing makes sense. This is the second school of thought. I'm actually part of the second school of thought, and I always believe that you need to have replicate. So probably Francis will touch more on the replicate side. So these are the two issues, and which is ongoing issue, and a lot of changes are happening in the algorithm as well as, and some of them has to do with the quantification, some of them have to do with experiment itself.
Yep. So two things Bonnie mentioned: the sparsity of the data, meaning the zeros in our data, right? So that's one of the issues here. They call it zero-inflated matrices, whatever you want to call them. That's the sparsity in our data. But I was saying that somebody was trying to do some calculations, and they were saying, oh, with the sequencing we do anyway, you can explain those zeros. 10x, though, says something a little bit different, which is basically, depending on the chemistry, that it's been evolving, right? They estimated, I took this from 10x because I always need to check what is the actual percentage. They're claiming that with this version of chemistry they use, basically you are capturing a third of whatever that cell is expressing. But the power, right, of single cell is you are not analyzing a single cell by itself. You have a group of cells that, in theory, is part of a tissue that you care, and then even if there's a sparsity, you may have enough information to do analysis because you have a lot of cells in that group, and if one cell doesn't express a gene that it should be expressed in the group, some other cells would do. Actually, there are methods to fill the gap of that matrix. Whatever you use them or not is a little bit up to you, but there's a method called magic, for example, that would fill the gap, those zeros, based on estimates of their neighbors. So if you have a cell and all the neighbors around it, they express a gene, they would say, okay, this cell needs to express it too. So let me fill the gap in your matrix. They do that in a way to improve the downstream analysis.
It's a random number, or it's like a mean of all those cells?
Well, I don't know the statistic behind it, but there are different methods of filling the gap of that matrix. Yeah. They first built, all of those is machine learning approaches, and they talk about manifolds and how the data is built, and they look at neighbors and then graph theory. It's the problem of, I feel more or less comfortable to talk about bulk and the statistic behind it. I'm starting to feel not comfortable talking about a lot of methods that are related to this because they are more related to machine learning approaches and graph theory that I'm not trained for.
So what does this 10x actually means in the single cell analysis?
Oh, 10x Genomics is the company who is one of the most popular companies doing single-cell RNA-seq. So it's just a company who builds the machines, software, and the chemistry for analysis of your single cell. We talk about large amounts of data. That's going to be an issue.
DropSeq was invented in an academic lab, and then it was taken over by 10x, isn't it?
The microfluidics, I think it was Again, it was Rain Dance or one company. I think it was a Bonnie. Bonnie knows those better than I do. Well, if we can ask him when he stopped talking. But yeah, I'm not that familiar about the history of who invented what. But yeah, I'm not sure where 10x took it from. Yeah, but a lot of those things, right? They're born in a lab, and then the company buys them. I mean, the whole business in San Francisco of idea of startup companies is that, okay, a startup company in 2 years, I'm hoping that Roche comes and buys me, right? And then I don't have to work the rest of my life, I guess. This is also another thing, right? That it's sometimes we don't think about it, but when we work in single cell, depending on our question, sometimes, most of the time is about cell types, right? I care about endothelial cells. I want to compare endothelial cells between sick kids and normal kids, normal or healthy kids. And I want to know the differential gene expression of those groups, for example, right? That's the type of questions. But we are working to so much detail that is biological variability that we don't care maybe, right? If the cell is dividing, the expression of that cell is changing. But I don't care if it's dividing or not. I only care if it's an endothelial cell, right? So there's going to be variability in our data that's biological that we may not care, or if the cell is dying, or if the cell is doing something else. So there's other things that we need to think about it that it may be important in the way that we do the analysis. That when, in theory, they are not present in our book, or we cannot do anything about it when we do book. Analysis-wise, there's a lot of commonalities in general terms, right? If we think about it, what we did yesterday, there's a lot of similarities, right? So there's some pre-processing of our raw sequencing. Bonnie talked about how we use barcoding and UMI, and I'm going to review that later. But we need to produce a count matrix. So the count matrix still exists for single cell, and it's the one that have a lot of zeros, the one that we mentioned. So we build it. So there's not much different in that sense with bulk RNA-seq. So we're going to get our reads. We're going to do some quality control. We're going to map it to a genome, and we're going to produce a count matrix. Then there's some QC after that about the cells. We may throw away cells because we think they are dying, because they are doublets, or other reasons. We're going to normalize our data. It's the same story. Our count matrix is not explaining everything. We need to normalize it somehow. This step is not present, or I guess I don't see the equivalent in bulk. But yeah, we may select some of the genes that are highly variable and use that for PCAs. We kind of did it yesterday, right, with a normal PCA. We selected 5,000, and we did a PCA with that. So same idea. We may choose the high variable genes to do our clustering or our UMAPs or TSNEs. Again, dimensional reduction, we're going to use it. It's not the same type, or we don't necessarily use the same type, but similar, but you're going to do dimensional reduction. The same thing in bulk, or similar things in bulk. Another step, in this case, we don't do, well, we remove batch effects, I guess. If applicable, I put it between parentheses, we can do integrate different data sets. We can have batch effects also in single cell.
Francis, can I ask a question?
Yep.
So in case of single cell analysis, what do you prefer? Like you want to have a complex mixture of cells? I'm just asking from a statistical point of view.
Yeah. Oof.
So
Okay.
will that actually, like you sort cell and make it less complex versus having a complex set of cells? What do you actually prefer? Is there any statistical advantage in terms of analysis, cluster differentiation when you have more complex mixture versus less?
I know. If I don't think too much about it, I would say there's not much a difference, right? Having complexity is going to change anything? I don't know. If you only have one cell type and you do single cell, you don't care about clustering, except if you want to do subtypes, right?
If I say
But
like I have 10 cells mixed and 20 between 20 different cell type mixture. So
I know what you're asking, like having variability in your data, right?
yeah, it will give more separation in the clustering and
If you give a single cell type to a single cell RNA-seq analysis, if you cluster, you're going to get clusters. It doesn't matter. It's going to do the clustering algorithm is going to do an effort to find difference and to cluster. Although you know the cluster biologically means nothing in that case. Or it means something in the sense that those cells have some similar expression and those ones have some similar expression. But at the end of the day, if you are working with endothelial cells, that's the only thing you care, you have a group of endothelial cells. Clustering.
Even within a seemingly homogeneous population of cells, for example, T cells,
Yep.
there will be subsets.
Oh, yeah. There could be subsets. I mean, it could be biologically important.
For example,
But
neutrophils, you will not feel like, or even monocytes for that
sure.
matter. There will be subsets there.
Yep.
It could be different.
Okay, take one subset and you do clustering, you're going to get clusters. Take a subset of the subset and you do clustering, you're going to get clusters. If there are some differences, they will find it, and clustering algorithm is going to make an effort. I don't know what point is going to stop clustering, right? But you even in a subtype, if you try to do a clustering, it's going to find some. The clustering, what I'm trying to say is, clustering doesn't mean cell type necessarily as a biological thing, right? We'll see. We will play with clustering later, or in the practical part of it, and you will see that you can change your clustering a lot. Right.
Is being done on what basis?
On the expressions, yeah. It's detecting gene
Genes in one.
Yeah, I mean, if you are assuming that your cell type is going to have some commonality in gene expression, and that's what the clustering is finding. I'm not saying, what I'm trying to say is that not always clustering is necessarily what you have in mind as a cell type, right? I mean, you can do clustering, and as you say, you can have a group of clusters that are T cells, right? But you have a group, because there are subtypes within T cells, and the algorithm is detecting the subtypes too.
Do you also do like something like site-seq where you combine like proteins along with?
Yep. Yeah. Well, I think that the field is evolving to that even more, right? So this is not something we're going to deal with it, but you can do multimodality analysis, right? In single cell, you can use surface protein detection, for example, to classify your T cells, also doing single cell RNA-seq at the same time, right, with the same cells. And the field is clearly moving towards that, like do proteomics and single cell at the same time, doing ATAC-seq and single cell at the same time, RNA-seq at the same time. So clearly, we are moving a lot more to that, to do multi-modeling at the single cell level at the same time.
So your antibodies are tagged to oligos in that way.
Yep. You have different systems, though, right? I mean, but yeah. So site-seq 10x, they have ways to do surface proteins detection, classification of T cells, and then at the same time you're doing so T cell receptors, BCRs, and all that. So those are available, I guess, in commercial kits. And yep, there's downstream analysis to this, right? You can do clustering, differential expression, cell typing, pathway analysis too. The pre-processing, I don't know if Bonnie talks about it. So I'm showing it again because there are two words that we use sometimes. The barcoding, when we say barcode, at the end of the day, it's the same. Well, it's the same. They are DNA sequences, but we talk about cell barcoding. That's the ID for the cell. And it's just a DNA sequence that we added through the process of library preparation to identify different cells. Then we talk about UMIs, which is also an tag, an identification tag, but is identifying original transcripts. UMI stands for molecular identifier, right? Unique or universal, I think, is a molecular identifier. We've been using this in the field of analysis, DNA sequencing for years. It's not new for single cell, right? But we use them.
Can anybody tell me why UMIs are used? I actually mentioned this in a different context, but in a single cell context, why they are using UMIs here?
Well, it's funny to me they call it UMIs, but it's a barcode, right? It's a random generated barcode.
So basically, this is a low input library. Single cell libraries are low input library, and there will be a problem of PCR duplicate. So effectively removing PCR duplicates, UMIs are used in this context.
Yeah, so when the software detects same barcode, same UMI in two different sequences, so there are two reads, you only count it once because it comes from the same molecule. It's been duplicated through the process of library preparation. It is not real data. In this case, we have the same barcode, but two different UMIs coming from the same gene. You know that it's two different molecules that produce this, so you need to count it twice. So that's the idea.
Can it be done that the cellular barcode and UMI, the length of that can be decreased? Because actually what you're sequencing, the insert is too, like you're sequencing more of the barcode and the UMI than the actual
Yeah.
sequence. So, I mean, if you could somehow,
I'm assuming they
the minimum that is required.
that's what I don't know. And they probably had redundancy too, in the sense, to be sure we had extra bases. I don't know if they actually did the minimal or not. I'm not sure. That you can probably calculate it, right? You can estimate how many possible molecules you can get, but also how many you can sequence. I'm assuming sequencing AAAA is not good. So that barcode or UMI is not going to be that useful. So I don't know how they estimated the minimum.
Sandeepji, the data here is the barcode and UMI, not the other side. Other side,
So I mean, like,
the whole idea is the other side you should have just enough.
like when we're actually comparing bulk seq with single seq, we should not use single cell in that sense for, I mean,
But I mean, the whole goal is just to identify what gene produced the transcript.
not to look at gene abundance.
Yeah. Well, no, what I'm saying is you can identify a gene by a sequence of 30 nucleotides. Maybe 30 is enough to say this fragment of RNA-seq comes from that gene. You don't need 150. I mean,
So in single cell, the identity of the cell is more important than the gene.
Identity and UMI, because the
I mean, the UMI is going to tell.
yeah, it's a prompting application. So it's what Francis is showing you, the collapsing of UMI is important. So the real data of single cell is the barcode and UMI. And the second part of the data
It
which
somehow feels a waste actually.
no, that is the data. Actually, you think from a bulk RNA-seq perspective where you are sticking indexes only to mask it. But here the data is actually UMI and barcode. And the other portion of data is just to map it back into the gene of interest that you are interested in.
so like yesterday, count matrix. So this is the main type of table that we're going to use for analysis. So the pre-processing, we are not going to do alignment or the initial QCs. Generally, or probably one of the most common tools is use 10x software called Cell Ranger for this. And Cell Ranger will give you already the count matrix that you can use for analysis. It's free software. Also, they do a QC already for you. They have initial QC, so you can see if your library is ready for downstream analysis or more analysis or not. You have alternatives, though. So you don't have to use Cell Ranger. Remember, we talked about bioconductor, the R. Surat is not for alignment. So after, when we have the data metrics, we're going to use Surat for downstream, for analysis. But before getting the count matrix, you don't have to use Cell Ranger. At the Genome Center, we run the Cell Ranger for everybody that comes. We pre-process the data for them. But I don't know if where you're going to be sequencing, they're going to do it. If they do it, good. If not, you can use Cell Ranger, but there are alternatives. In Bioconductor, you don't have one package that does everything, as far as I know. I don't think that exists. But they have like documentation on how to combine different packages to do this whole process. So if you want to work in R, I don't know if you want to, but you can do the whole process in R using Bioconductor packages. And they have what they don't call it best practices. They call it something else. I don't remember. But they have documents designed to walk you through the whole process and use only Bioconductor packages to do the whole analysis. And they have it for spatial too. But that's not using Surat. Surat is not part of Bioconductor in that sense. So yeah, Surat. So I would say probably is one of the most common tools for when you have the count. Surat would be the same as DSeq2 for single cell. So the rest of the steps, and this is what we're going to do today. So we are not going to do the alignment as we did manually for bulk RNA-seq. is to work within CIRAD and do all these steps of the analysis that I mentioned before. Yeah, questions? Oh yeah, the alternatives. So if you like to work in Python, probably the most popular one is ScamPy. If you like to work in Python, you can use ScamPy for analysis. And what I said, using the bioconductor ecosystem, because again, I don't think there's a package that would do everything at once, so it's a combination of packages. And again, you can consult, I forgot the name now, how they call it. It's called OSCA. If you Google OSCA, that it means, I don't remember what it means, it's single cell analysis. I forgot, orchestrated single cell analysis probably. Bioconductor, you will get a document of how to do single cell analysis using bioconductor packages. And they have the equivalent for spatial. I'm not advocating for one or another in any sense. We work with CIRAD, and we stick with CIRAD. That's the only reason. So what type of, I'm going to go a little bit through the steps now before we go to the doc for the Jupyter Notebook, just to review them. So we're going to do some QC process. So one of them is to remove cells that don't have enough sequencing, right? So if they don't have enough sequencing, they're going to be noisy. They're not going to give us enough information to do analysis. I think this is pretty obvious. Amit mentioned yesterday, Dr. Amit mentioned yesterday the idea of we generally remove cells that have high mitochondrial expression. I don't know, there may be cases that you care about mitochondrial expression, then you may have your reasons to not remove it, but it's quite common that we remove cells with high mitochondrial expressions because we are assuming that it's biological noise. I was mentioning earlier, right? That there are cells dying. They're doing something else that we don't care about it, which is dying and not being helpful for our analysis, or being super stressed.
So what about if you are working on apoptosis or necrosis?
Yeah, that's what I said. I mean, there's a lot of things that probably we don't know enough of those cells that have high, and maybe they have 20% of the expression is mitochondrial. I guess maybe we don't study them enough, and there's good reasons that they are there doing something, right? But yeah, that's what we do. We remove those that have high expression of mitochondrial genes because we assume they are dying. We're going to try to remove batch effects. We'll see how much time we have. If not, we can talk about it, how we can do it. So I said remove again. It's not exactly remove. Like with DSIC2, we don't necessarily remove. Modeling, I don't know if that's the word. I don't know. We do something about it. I mean, there's different technical effects. We're going to go through it. We talk about that also. We're going to detect some ambient RNA. Actually, part of this ambient RNA is already removed through Cell Ranger, right? So it's going to be removed most probably by the time we get it. And I think we mentioned this at some point, right? We talk about doublets or multiplets. That is having two or more cells being captured together. I always, it is not a bad number to have in mind, in the sense the expectation, I think this is based on 10x data. So this is an important thing. I'm assuming that everything we're doing today is through 10x. There are other ways to do single cell library preparations or techniques, right, to obtain single cell data analysis. But I'm assuming what we're doing is using 10x. 10x, and also depends on the version they're using. I forgot if this is the latest version or not. So it's an estimate of 4% of doublets per 10,000 cells that you're analyzing. And that is linear. So if you try to analyze in a single library preparation 40,000 cells, you need to multiply by 4, so it's going to be 16%. Oh, this must be the latest one, actually. This is the latest, because it's very low, right? So let's assume you want to remove doublets, because you care a lot about removing doublets. You will see that you may not care that much, but let's assume you care, and you want the cleanest data possible. you need to assume that if you have 10,000 cells, you estimate you need to remove around 4% of your cells, because those are doublets, right? So it's a good number to have in mind.
So if you have not enough cells, can you decrease that? I mean, like, can you
Yes. So I think also it's even myself, I found myself sometimes not filtering enough. I think, why I'm not filtering enough? I have 10,000 cells. Do I need them? Maybe not. Maybe, I have a cell type that is abundant enough that I don't need 10,000 cells. I can filter them really harsh to get the highest quality possible and get a good analysis out of it. And I don't need all 10,000 cells and make an effort of saving 100 cells. Oh, I feel afraid of removing 100 cells. Don't be afraid. If you have enough cells for the experiment, be really harsh filtering. Why not? Yeah, so that's the problem. If Rick Pierce is working with only 20 or 30 or 40 endothelial cells, you need to conserve them as much as you can, right? And be aware that maybe the quality is not that good for those. Even for normalization, right? So when we filter cells with low expression, those are going to be problematic. Why don't filter even harsher and you get all cells to have similar expression level or similar sequencing? That's going to help to the normalization process. If all your cells are really narrow in a space of how much sequence you did to them, the normalization is going to work great. If all of a sudden you're working with cells that have only been sequenced 500 times and cells that have been sequenced 10,000 times, the normalization process is making an effort to normalize them, and it's going to be noisy no matter what.
And what about, so I believe the doublets are recognized using the RNA content.
There are so there are two ways to do it. We're going to use the simplest possible, and I'm not convinced it's that great, because when you use so let's assume you use these packages that are going to make an effort to detect doublets. So there actually are packages that are designed to detect doublets only. The question is if they work great or not. I don't know. I don't have enough experience to tell you. But they do detect doublets, and they seem to detect doublets at the fraction that we're expecting. But when we use I'll go through that. But then let's leave it here. If you want methods, specific statistical methods, I guess, or algorithmic methods to detect doublets, you can do that. There are packages dedicated only to that. We are not going to do it. I decided not to do it because maybe the normal way or the simple way to do it may be good enough. But I mean, they exist, and not to add complications, I guess. So I think I already mentioned some of those things, right? So we're going to do normalization. Seurat provides two methods. Log normalize, which would be a really classical way to do it. You always need to divide by the size of your library, and you log transform them to get narrow, to avoid the extremes. And we talk about the idea of log normalizations for bulk RNA-seq. They have another method. They call it SC transform or SCT. We call it SCT. I'm not even sure if that's the correct name, but for short, SCT transformations. That's more recent for Seurat, but it's also available. This is what they recommend, by the way, to use, but we're going to do both. We have some process of selecting variable genes, so those are the ones we're going to use for clustering and building our UMAPs. The process of dimensional reduction, right? We talked that we do PCA for bulk RNA-seq, basically because it's assumed linear. That doesn't work for single cell because the data is not assumed to be linear. So the cells, the expression of the cells are not linear. They talk about manifolds, how the cells are in a different space that it's not linear. So they use different techniques and, again, graph theory to build these types of dimensional reductions. If you work with single cell before, you heard about it, you probably heard about UMAPs and TSNE. PCA is still used. PCA is always a way to reduce because we work with such a big matrices, 10,000 cells, a lot of genes. So there's ways to reduce that information and keep as much information as possible is using PCA. Actually, UMAP, one of the first steps is to do PCA, and then uses the PCA, the principal components, to do UMAP, the whole process. So PCA is used, no matter what, at least internally, to do the dimensional reduction. CIRAD also offers a ways to integrate different batches. So if you're doing replicates, right? We talk about replicates. Oh, another question that we need to clarify with the replicates. So if you're working with actual single-cell replicates, you do your experiment with multiple samples, right? Then it means you're going to have 10,000 cells, for example, one, 10,000 cells in another, 10,000 cells in another replicate. So sometimes what can happen is that, and I'm going to give an example. I'm not going to talk much, but there are ways to reduce possible batch effects that can happen during single cell. There are different methods. Actually, now CIRAD, I don't know since when, recent versions, they keep offering new methods or more methods to do integration. And again, we're going to do some clustering, identifying cell typing, differential expression. Those are more similar to what we do with bulk. When you work with replicates, right? So if you think about it, if we work with one sample, we can consider every cell being a replicate within that cell type, right? So what happens when we get another single cell that is a replicate? So two samples, we process two samples that are replicate of each other, and we get in 10,000 cells of each. So the analysis changes a bit, because all of a sudden you have, I don't know, I'm making out this number, 500 T cells in one, 500 T cells in another one, right? So you have two replicates. So one way to do an analysis is what Bonnie was saying, is saying, let me do pseudo-bulk. Let me convert this into a bulk data. So I'm going to get the averages of all the T cells in one sample, the average of the T cells in another sample, and then all of a sudden we convert that into bulk RNA-seq, and the comparison becomes easier than if we are working with 10 cells on top of that. I don't know, the analysis is going to get more complicated because the simplest way to do this is to become pseudo-bulk and then do comparisons if you're working with bulk, if you're working with replicates. A lot of people doesn't work with replicates necessarily, right? So they come with one sample and they do their experiments. Generally, it's an exploration process of saying, okay, I discovered that this gene is really important for T cells under my treatment. They go to the lab and they do experiments. So it's their own risk, the way they do the analysis of saying, oh, the gene that I discovered within these test cells is important for my biology. And then they go to the lab and they do the analysis to validate it, right? So it's up to you how you use these techniques for discovery in that sense that it's important to use biological replicates. You want to do them because they're going to give you more sense of security when you go to the lab and you do experiments in the lab that you selected the genes that are actually important in your biology.
This variant stabilizing transformations that you use in single cell is same as what other institutes or
No.
different?
There is slightly difference, but the idea, I don't know behind the scenes how they work, the details, but the concept is similar, right? Definitely they don't use the D-seq approach. It's not, you don't use it. You're using log transformations, you're using variance stabilization. We talk about the idea of variance stabilization to the point in SCT that you don't have counts anymore when you do differential gene expression analysis. So you need to stop to think about when you, in the latest stages, when you're doing D-seq2, you are not comparing counts. And we will talk about that when we do the analysis in the Jupyter Notebook. You can actually use D-seq2 inside of CIRAD. I don't necessarily recommend it, but you can use it. It gives you the option to do, when you compare cluster 1 with cluster 2, to say, okay, you can use the D-seq2 model to do the whole analysis of the comparisons. I don't necessarily recommend that. It's super slow.
I was thinking like that. My understanding was like, maybe wrong, maybe, that the pseudo-bulk differential gene expression is done through D-seq. Not true?
Sorry?
The pseudo-bulk analysis done in single cell is not built on D-seq2?
You can use D-seq2 for pseudo-bulk, yes. But we don't do pseudo-bulk with one sample. If you have one sample, the analysis is assuming, right, that your cells are replicates. That's the whole idea, kind of. Yesterday we talked about the DC2 object and how we access data into it, like we do the coefficients, we ask for results, and all that. And it's relatively complex, so the CRAD object is even more complex if we compare it with yesterday. So we're going to have cell metadata inside of that object. We create a single object, right? In our code is going to be a single word. And that single word is defining an object. In that object, we're going to have all this information. So it gets a little complicated to access it. I mean, we will see how we do it, but you're going to have a metadata in the same way we have the metadata for our samples. Our samples now are cells. So this metadata data frame is huge, right? Because we have a lot of cells. And we can keep adding columns and columns and columns and columns to our metadata. We're going to store also our dimensionality reductions, our PCA, our UMAPs, our t-SNEs, or whatever method we like to use. It's going to be stored in the same object. There's going to be all. Sorry, with the I don't know if it's the fan or something. I can hear you. Yep. Oh, totally. Yes. Yeah. It definitely does. Yes.
How many times? How many more?
So, okay. So if yesterday, I think each one of us.
I'm asking them because then they can do in 2 or 3 batches so that it doesn't crash at the end.
No, it won't crash. If you want, we can talk about it. If somebody cares about AWS, I can tell you about my experience, and we can talk about it. So yesterday, we were using 1 gigabyte of RAM each of us. So just to have an idea, we used 40 gigabytes of RAM. So the computer that we had more or less had that. So we know it wouldn't crash. So for today, we're going to use 10 times more that. No, actually, close to 12 more. So it's going to be 12 gigabytes of RAM each of us. So multiply 12 by 40. So we need 500 gigabytes of RAM computing power. I don't know. My computer may have 16, right? So you can imagine how much power you need. So with a laptop, or at least my laptop, I'm assuming if you have a newer laptop, maybe you have a little bit more. But nowadays, a laptop has around that, 16 gigabytes of RAM. You can do a single-cell RNA-seq analysis of what? Maybe 10,000, 20,000 cells. If you're working with 100,000 cells that we work sometimes with, forget about the laptop. There's no way you're going to work with it. I think the biggest analysis, single analysis, I mean, some people in our group, they've done really big ones. The big one that I did, I was using 300 gigabytes of RAM for the single analysis. So it's clearly single cell, you need more compute power. There's no escape. Probably one important thing is the count tables, because we're going to have multiple count tables inside of the main CRAD object. One is going to be the row counts, normalized counts, and what we call scale counts. And not only that, we're going to have that before and after normalization. I always get mixed up the names. They call it layers. So the CRAD object is going to have also layers, pretty much duplicating all this information. And do you care that much about the internal parts of CRAD? I think just to be familiar about the names a little bit. But I think once you start practicing, you will see how you access it. You can think about columns maybe or things inside, and I'll show you how you access that data. It's not that complicated. But you need to be familiar that it's not just a table where you're processing. It's an object with multiple tables inside. I think I mentioned that already. I don't know if I should go through this, but that is the main idea, right? That t-SNE and UMAP are actually the methods we use for to show the dimensional reductions. So this is how it looks like a UMAP. So every dot is a cell, and this is the dimensional techniques that we use. So the most common thing is to show 2 dimensions, the 2 main ones, right? Like PCA, but you can get more dimensions if you want to. And nobody thinks about the 3rd dimensions in UMAPs or t-SNEs, but they do exist. And the reason is because, again, the way that the cells interact, or the space of the cells in single-cell RNA-seq, the data, is not represented by some linearity process or anything like that. They describe it as manifolds, and then is when I'm not going to explain what a manifold is because I'm not going to do a good job, right? But yeah, so internally, we are using PCA to reduce dimensions first, and then we use the UMAP approach, for example, which is the most common nowadays. Disney are starting to not being used that much. We tend to use UMAPs more. And I can tell you, well, there's a reason for that, although, again, there's a lot of discussions of why one is better than the other. But so, yes, we group cells by clusters when we do UMAPs. And we assume that a cell next to another, it means they're close. What about the white space? So is this green cluster here far away from that green cluster down there? If you look at the UMAP, you can think yes. Well, we know that's not that true. So the white space between these can be meaningless. There's no distance between clusters. UMAP is not designed for that. That's the claim. Sorry, Disney is definitely not designed to evaluate how far is one cluster from another, right? UMAPs claims that is a bit better on that sense. Then later on, people saying, no, that's not even true with UMAPs. So be careful. Don't try to make interpretations of, oh, this cluster is far away from this. It means it's not related at all or whatever. So white space, it's...
Why they have not just put all these clusters together like?
Sorry, what?
So if the distance is not useful, why they have not put all the clusters together?
Why they don't do it? Oh, it's part of the calculations, I guess. I don't know. And actually, again, remember we talked about setting the seed, set seed, and we give him a number. So some of these processes do start from random seeds to start the calculations. So if for whatever reason the random seed changes, you can see slightly variations in your data. Like suddenly this cluster is here when you expect it to be here. But again, it's not that important. Another important thing that if I don't, now that I remember, I need to tell you that now, probably I may forgot later. So I'm talking about a cluster. Clustering process and UMAP is different. First we build the UMAP, then we do the clustering, and then we overlap the information. So this is green because I overlaid the information of the clusters. The UMAP is not telling me anything about the clustering processes. The expectation, though, is that there's going to be clearly an overlap, right? That the UMAP, the dimension reduction of my data is also conserving what the clustering process is telling me. But it's not necessarily true. Why? Because sometimes we may have what it looks like a big cluster, and it's divided in fragments by the clustering processes. Dr. Amit, I think, explained these a little bit, right? I think somebody mentioned it, the trajectory analysis or pseudo-time analysis and all those things. You can do them also in single cell, right? So you can, well, basically it's a cool application of single cell, trying to figure out states of a cell and how they evolve. And what they call a pseudo-time, you can color cells based on some gradient of time, early and late, for example, right? And study the evolution of that. There are a lot of different packages. Some of them you can use them directly in CRAT. Some of them you need to go back to the sequences because they are based on the idea I have an RNA-seq sequence that hasn't been processed yet. It hasn't been spliced. I'm going to call that early stage. And then I see it in a different cell is a completely processed. That's a later stage. Right? So you may have packages that you need the sequences to build the pseudo-time process. There are others. I mean, trajectory analysis generally work in clusters. You can tell this is the original cluster, and if we try to build a trajectory, right? Pseudo-times, you can call, although really similar and overlapping, they talk about cells and how color the cells have some sort of projection of time. Yep.
So are there any technologies where you can do it on a long read platform like Bioinformatics?
Oh, I've never done it. It's an interesting question. I guess if you want to study what we said, right, pre-process and post-processing of that same RNA-seq, that could be the same transcript. That could be interesting. I don't know if it's been done. Oh, clustering. OK, good. I didn't know if I had a slide about clustering or not. I have it. Good. So generally, the main goal is to that, right? To identify cell types. We're generally going to assume that we have the tendency to assume that one cluster, one cell type, right? That's going to be our general assumption. It's not necessarily true. It could be more things than that or less things than that, right? Clustering is important because it allows us to identify units in our data that we can use for differential gene expression later, right? We can say, I want to compare cluster 1 with cluster 0 to know what genes are different, right? And also, there's different methods for this, for types of clustering. But importantly, again, UMAP is a different thing than clustering. They're two different processes. But we tend to show both of them together. So we show the UMAP, and we overlay the colors of our clusters on top. And the expectation, of course, is that it's going to be a lot of overlap. You're going to see, I don't know if that's here. Oh, yeah. I don't know if you can see it. There's a green dot in the middle of the blue dots. So the clustering is telling us that this green dot belongs to cluster 4. The UMAP is telling us that this dot is closer to these ones, in theory, right? So this is going to happen all the time. Maybe it's a doublet. I don't know. Maybe it could be, right? That could be an interesting exercise to do to figure out, OK, those dots that are not, like we have another dot here that belongs to this, but it goes with that. I don't know. Those could be maybe doublets, and it's detecting the signal of both. I don't know. Could be a good exercise.
Similar expression. Or those two cells could be interacting with each other?
No, if the interaction, I don't think so, because interaction generally we evaluate it. You can think about expressing a receptor and expressing the signal of that receptor. They're going to be two different genes being expressed, right? OK, batches. So I forgot that I have a slide about these two. That's good that I have it. I think this is a real example. And actually, it's an example that if we have time, we work with it. I think it's the data that I use for the workshop. So in here, we have two samples that we are expecting are going to have the same cell types, right? When we do the UMAP and we color by sample, you realize there is barely any overlap. But we know they're the same cell types. So there's no overlap. That is indicating that the difference that we're observing, or some difference that we're observing in this data, it is caused by some batch effect, not by real biological data. Again, I'm going to quote not real biological data, because this is dangerous, but I know that we am expecting some overlap, because there are cell types. I didn't do anything especially different between them, so I'm expecting some overlap. And I want to identify T cells in both of them, for example. If I try to identify them when there's no overlap, it's going to be harder, because I'm going to have a T cell in blue and a T cell in red. I'm going to have monocytes in blue and monocytes in red. So there are processes that what they do is you may try to remove batch effects, or you may try to find commonalities and overlap them. So it's easier to do downstream analysis. If we have time, what we're going to do is to overlap them without modifying the expression. We're just going to overlap them so we are able to identify the the cell types of interest, and then do analysis later on. But this can happen. Now the question is, do you need to do batch correction every single time? I'm more in favor of evaluating first if you need to do batch correction, and then do it if you need to, or not doing if you don't need to. So if you don't have this situation, I wouldn't do batch correction. Another way you could look at this is analyzing every sample separately, identifying your cell types, and figure out a way to merge the analysis and do the comparison. You can label them first, right? You can figure out what cell types you want to work with, identify them, and then do the label them and merging. If you're working with a lot of samples that have been processed in different time points, at that point, it's going to be hard to evaluate how much overlap you have or how much or not have. So then I would recommend probably you don't have an option just to batch correction every time, right? But if you're working with 2, 3 samples, it's easy to evaluate how much overlap you have. Cell type annotation. Different approaches. And this is also evolving fast, too. We see new packages, new things that help you with the cell annotation. There's a possibility of doing manually, totally, and I think it's great. It's a good one, right? You can do manual annotation, meaning if you figured out genes differentially expressed in every cluster, those are going to be the ones that identify that cluster about something, right? So if you can identify genes that are only expressed in T cells, then you can classify that as a T cell type, right? So, oh, I have no idea.
There are a lot of hepatocytes.
Oh, yeah, probably.
Even the hepatocytes, they are like different clusters. And some of them are very...
Yeah, they just number them. Yep.
Yeah, but one of them is up there, close to the macrophage.
Yep. Yeah, again, distance
Doesn't
doesn't mean necessarily means anything here. It's dangerous with UMAPs, right? UMAPs claims that it does to some extent. Later analysis, there are papers out saying, no, it doesn't. So it's always a bit dangerous if you try to do those type of correlations. Yeah, but again, don't worry about it, how they cluster here, right, that much. But yeah, you can have these situations.
How about the relative position of these clusters? The relative position?
The relative position of the cluster in a UMAP, so how much white space you have between them is meaningless. Or you need, my recommendation is you need to assume that it's meaningless.
differently, they must be having a different set of genes.
Yes, yes, yeah, totally. But even here, these ones have a different set. It's what I was telling you. Depending on how you set up your clustering and everything, you may have these as a single cluster or as a multiple cluster. Depending on your settings, depending on the arguments that you use, you can change the number of clusters, you can change the resolution. Yeah, but I mean, clustering and UMAP are two different things. So you need to have that. Yes, of course, the UMAP is based on the expression, the clustering is based on the expression, but both processes are different. They don't need to completely overlap necessarily. And of course, we can do differentiation expression analysis. We can make our enhanced volcanoes and so on. So this is common. We've seen them from yesterday. We talk about data formats. I'm not going into details of these ones, but Cell Ranger, if you use Cell Ranger, it gives you two options to choose what input you're going to use for your analysis. You can get the raw matrix. It gives you actually multiple files. It gives you three files where you're going to have your raw matrix there. So similar to like we did yesterday, we had a table, but these are going to be much bigger. Or you can use what is called an H5 data format, which is even richer. You can have more information in an H5 data format. It's a single file, and you can process it in R as a single file. And here we are reading a folder with 3 files in it. But it doesn't make a difference for the things we do, pretty much. So any option is fine. For the example today, we're going to be using the H5 file. And I don't know if maybe I kept as an example also the same data stored in a different format. I don't remember if it's going to be in the Jupyter Hub or not. And I think we're going to be ready. When we have the tea break? Oh, great. So I have time to start the Jupyter Hub.
So we'll break for 10 minutes, like 15 minutes. By the time, I think the AWS will be on.
Yep. So same gene expression profile would be that you don't see differentially expressed genes, for example, right? So if you have two samples that you barely see any difference, that means they have the same gene expression profile. Having a lot of differences would mean they don't have the same gene expression profile.
Cell cluster, one cluster will represent the
Hello? Oh, it works. Okay. Oh, then. Okay, good, good. So is where are the speakers? There are speakers in the back there? Oh, those big ones. Okay. But not in the back.
Hello, 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 hello.
Should I start? See? Yes. And now we're speaking Spanish.
Yes.
So you have the day 4 folder. I mean, you know the drill, right? So inside, there's 2 subfolders or directories. One that says PBMC. That's the one we're going to use. There are 2 data sets. So the first one is a PBMC, so blood cells. The second one is PBMC too, right? But with one patient of, it has this type of cancer called multiple myeloma. We're going to start with the one, the PBMC. I think I got both of them from the 10x Genomics website. So they have a lot of data sets. So I picked 2 from there, and that's the one we're using for analysis. They have from the raw sequences, if you want to work with the raw sequences, up to the count data, right? Or our tables, or the count matrix.
Is that data freely downloaded?
Yep. So everything we're using today, both of them come from 10x Genomics. And they have even, they've been accumulated different versions in the sense that when they use all chemistry, more recent chemistry, and super new chemistry, they have different types. So if you open that folder, you're going to have the notebook that you're already familiar with, the extension, and the H5, that file that I mentioned, that contains the count matrix and metadata. And I also added this HTML that I'm going to open now. You can open in your computers too if you want to. It is blank on your side too?
Yeah.
Oh, trust HTML. Thank you. It must have some code in it. This is the report. I'm just showing one of, you can get even more complex reports, but this is one that you can get from Cell Ranger. Because I want to talk something that I forgot to mention during the talk. You're gonna get, you see these big 3 numbers? That's the one of the first things you're gonna want to check in your data. You're gonna get an estimate of number of cells that you have in your data. They give you, normally, right, I think the current version, they consider you can more or less load, they recommend loading like 20,000 cells per library, right? You can even do multiplexing. So we talk about barcodes per cell, UMIs per transcript. You can have barcodes per sample too. So you can multiplex in a single library multiple samples if you want to. So those 20,000 cells, they could be multiple samples. Well, but in this case, this is probably a bit older chemistry. We have 10,000 cells. This is probably one of the most important ones, right? This is the number of reads that on average that we get per cell, right? So this is what it's telling us how well we sequence. So you can call it the X factor, right? The 10x, the 30x in whole genome sequencing and so on, right? So this is the coverage that we get per cell on average. They recommend, this is a recommendation directly from 10x, that you should estimate around at least 20,000 per cell. So we have more than the recommended one. The more you sequence, the more genes you're going to detect. I'm not saying that is necessarily better, but you could. And the same concept we have about the curve of saturation. These 10,000s, they're estimated using this type of curve. If you scroll down, you're going to see it. So this is how they remove the background noise, what they consider background noise when there's no cells, when they detect some sort of big drop on the UMI counts when they count in barcodes. They sort them from the number of barcodes they detected and how many UMIs they detect on per cell. And when they see the big drop, they say, okay, here probably all this is just background noise. It's not a real cell. And you get a lot of bunch information that you can look at it. I mean, all this can be important to you too, right?
How do you define
You don't define, it's It's an algorithm internally who decides to create the cutoff. When it sees the drop in that plot, it says, okay, from here on, this is probably background noise. You can.
That read indicates UMI count will indicate.
The UMI counts is the number of transcripts.
So that is the identifier, right? The identifier of the gene.
Of the transcript, yes.
Yes. So if it is less than 100 or.
See, it's been sorted. You notice that the plot has been sorted, right?
Yes.
From the one that has the most to the least. And when you see the big drop, they consider that most probably everything else is just some transcripts that are floating around and they got captured in the bubble and they got sequenced. Or viable cells. It could be a cell exploded or whatever and liberated everything. That's how they consider it. I mean, there's a lot of things you can tweak when you do your analysis, right? Or you process the data with Cell Ranger. You have other data here, like how many reads map into the genome and so on. So you're a bit familiar with this. I mean, you can explore it. A lot of times, even though we tell you we run a fast QC and we do blah, 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 sometimes we don't, right? You only check it once you see there is a problem, right? So you do your analysis. Oh, I have a problem. I don't have enough mapping here. Why I don't have good mapping? Because there's a problem earlier, and then it's when you go backwards.
So are these the parameters which we need to check whenever we are running the single-cell RNA sequencing
Yeah.
and we are getting the data?
Yeah. So they already, I mean, they know how to tell you, right? I mean, how to give you the data. The big three numbers, check them, right? To be sure that your expectations are met. That's the first thing. Then you can check all the type of data down there. You can be sure that they even build saturation curves now, I think, in the new reports. How much saturation you get. Are you expecting to see more genes if you keep sequencing?
So my question is, like, right now we are seeing that the graph is still 10K of the cells, right? This
Yep.
The dark blue. So if it is less than that, that means the cells, estimated cell number was less, or that much have not been sequenced.
Yes, it's not a problem, right? Even though you're expecting 10,000 cells and you get 5,000, that's fine, right? I mean, it didn't work as you expected, but you still have some data that you can use. Meanwhile, this number is good. Meanwhile, there's nothing else down there that could throw your analysis out.
Is there any mean this much it should be like any number? Or
yeah, 20,000.
20,000.
That's what they recommend. You can use that. I mean, if you get 19,000, you're going to throw away your data? Probably not, right? But that's a recommended, above 20,000. I've done even analysis for less. But again, it depends. Are you exploring? Is your first analysis? Is it worth it not to throw it away? Just check it. Or not to add more sequencing. You can keep, if the center that did the sequencing keeps the library, you can add even more sequencing to it. The problem is why, sometimes what happens is we sequence enough to have 20,000, but you don't get 20,000. Why? That could be a problem, right? Maybe because we have a lot of cells that are dying and you're losing sequencing in cells dying or cells that are not real. So you need to evaluate why this is happening too, why you didn't get the expected one. The problem is with this, because it's expensive, we tend to be too tight to 20,000, and that's annoying. Because then if one sample doesn't work, all of a sudden you need to repeat it or add more sequencing. And that's always a problem for us too, because I think it's not plugged in, right? So, what I was saying? Oh yeah, so we can recommend, if you come to the genome or go to Genome Center, that, do an exploration analysis first to be sure that your data looks good. We're going to sequence less. And then they give you all the data back and say, oh yeah, you sequence less, but I'm not able to do an analysis or much with it, right? The only thing I can check is that I have a convincing number of cells maybe. Although this number also depends on this and how well you build this curve.
So one more question. Is it like 20,000 reads for every kind of cell, or it will vary depending on the cell type as well?
No, that's generic. I mean, there's no way, you don't know exactly. I mean, you probably, I don't know, you may be in a situation where, I don't know if that's even possible, but you're expecting low transcription, but you still can sequence very high. But I don't know, it's always hard to evaluate. PBMCs, you have everything, so it's pretty normal that you at least need 20,000, right?
So it's like the median genes per cells into the coverage, which is
This?
Does it convert into the mean reads? It means the coverage, right?
So see, this is the coverage. So this is literally this.
So if we have like 2000 genes per cell and the coverage is only 10x, then
Oh, you're trying to calculate the X coverage? Yeah, don't worry, it's not the same. When we say 30X coverage, when we talk about how many times you see a base in the genome, this is completely different. You don't have to think in those terms. I was trying to find an analogy when we talk about coverage, right? So we look at cell level only. So you don't estimate about any kind of genomic coverage or anything like that. Okay, so the data looks good, enough to do an analysis. So now you can open your notebooks, which is the one here. And I'm following the same pattern than in the other classes. You have a lot of documentation here that you can follow. Oh, I can see, maybe it scrolls too fast. These are the steps we're going to do, but I already told them in the PowerPoint presentation. In the same way that I told you Bioconductor is excellent in documentation, Seurat has pretty good documentation and tutorials. The main issue of their tutorials, they are chunks of information. They are not like end-to-end tutorials like you can find in Bioconductor, and that can be annoying because they answer specific questions, but if you want to do a full analysis, I found it, personally, I found it confusing in the way they build it, because the integration of all those pieces of information is not immediately clear. But there's documentation. There's a lot of documentation. And I don't know if you use GitHub. GitHub is a repository for coding, for code, for programs. They also have a GitHub page. And you can report issues. And a lot of people report issues, and you can read them. If you find the same issue, you go to GitHub, and you may find an answer why you see that problem. So yeah, don't be afraid to click the links, and you can see the website. So you already know what we're doing here, right? So we are requesting some libraries. We're loading them. So by default, R comes in some vanilla form, right? They don't have everything available there, but we're going to use these tools. They are already installed in Jupyter Hub. So we are doing here is, okay, let me get Seurat that I know is installed. Let me go all these packages. Let's skip this. These first three are part of Seurat. We need them. Well, we don't actually need them all, but let's say we can make use of them. Tidyverse, you already know them, is the one that we use the pipes, right, to create Excel files. This one here is for, these two here is for cell types classification. This is, again, is a library. So Seurat makes use of this for the transformation, for the normalization. I mentioned that the SCT transformed during the slides, so we need that. This is for plotting when we create ggplots. Oh, by the way, so Tidyverse, it's a set of packages. It's not only one. It's ggplot2, another set of packages. But it's really convenient just to load one, and you have everything you need in there. These are going to be tools that are going to be used. Enhanced Volcano, you already seen it. This one here it's complementary to ggplots. It allows us to create labels to indicate cells or things we're going to indicate in the plot in a nice way. Actually, what happens sometimes is when you load Tidyverse, it loads a bunch of packages. Same thing with Seurat, it may load other packages that we don't indicate here. It may be that ggRepel, we may not need it because it's loaded by something else, I don't remember, but just in case, I place it here. And forget about the options things. We needed to manage some internal packages about the memory RAM, how it's managed. You may not need it in your analysis if you do it.
You already loaded these,
The libraries are installed. We are loading them now, if you want to think in those terms. So again, you're going to see the typical, what we see every time we do the library thing, right? I forgot. So I don't think we have I think I removed the matrix at the end. So we had the folder. Remember I told you there are two ways to load the data into Seurat. One is through a folder with a matrix inside of it, like a real matrix that you can open and see the counts, or H5. I think I, at the end, decided not to include it to create problems or more, confuse people. It seems I keep the code, but I remove the folder. So this is nothing, so don't worry about it. You don't have to be worried. You can remove it or leave it. It doesn't matter. So what I'm doing here is indicate where do I have my H file. So I indicate in what folder it is. It's in the current folder, and it's called like this. And I store it in a variable that I'm going to use later to create this object, which is reading the file. That's the only thing it does. And the next step is creating the Seurat object. We're giving the data that we just read. We can give it any project name. It doesn't matter. It's just to have a project name. And we are doing already a first filtering process. Actually, Cell Ranger, the data that I'm giving you already is being pre-filtered by Cell Ranger. Cell Ranger is going to give you two H5s, I think. Again, depending on the parameters how you run it. But it's going to give you two H5s, one that is raw data and one that is being pre-filtered. The pre-filtered data is very lax in the sense that it's not being pre-filtered that much. So you are fine just taking the pre-filter if you want to. What we're doing here is we are saying to keep a cell, or to keep a gene, sorry, it needs to be expressed at least in 3 cells. If not, throw it away. If at least 3 cells express it, keep that gene. If not, throw it away. So what do you think we are asking here? Features, genes in this case. Yes. So yeah. So a cell, to keep it in the analysis, the cell needs to have a list. No, you're right. Sorry, transcripts, right?
Genes or transcripts?
Transcripts. So UMIs. Yeah, sorry, not genes, transcripts. So it needs to have a list, 500 transcripts detected, or UMIs. UMIs. So transcript is not the right word here. Well, no, it is. Transcripts, yes. It needs to have at least 500 transcripts to keep that cell. If not, we throw it away.
So is there any cutoff that has to take in this much only, or less than that, or more than
No, this is part what we said before. If you have enough cells and you only care about having the best data possible, increase those numbers.
So depending on the type of, how much cell count and the reach we are getting?
Yep. So there's a lot of iteration process in bioinformatics, even if you don't like it, that, I do an analysis, I see how the data looks like, I'm exploring the data, and say, I should change my model in bulk RNA-seq because I see that. So let's go back, change the model, do analysis. Same way here.
So one question. The output file is the PC matrix file, right? The file that comes from the
This is the file that comes from the Cell Ranger program that pre-processes the files. That has the alignment and QC for you, right? So these are the count matrix. Yes, what it tells you. I filtered, by the way. The file already told that I'm taking the pre-filter one, which it's pretty much taking these values, if I tell you. I think it's maybe it has a 200 here. So we are filtering a bit more than whatever they do. And by the way, do you need this here? Maybe not, because we can filter later. But we just given some numbers to have a smaller data set. So if I just indicate the name of the object that we created, our Seurat object, it's going to give us some small report. But if you notice, the original Seurat Ranger report, it says that we have 10,000 cells, right? So we went down maybe 100 doing some pre-filtering. Well, this filtering here, right? And we are detecting 22,000, in this case, it's genes. I don't know why they call it features. I don't think it's features in this case, but I need to double check. I think this is transcripts. This is, remember I told you I have essays on top. So the object that I showed you earlier that has metadata and count tables and all that, they have two types of duplicated data to some extent. Currently only contains what we call one assay, which is the RNA one here. When we transform the data, we're going to have two assays, the RNA and the transform assay. Each one of those have three layers: the count, I keep forgetting the names, the scale data, and the transform data. I don't know, we'll check it. But it has three levels to make it even more confusing, how the Seurat object and the complexity of it. The only thing I want you to remember basically is that for now we only have one assay. We haven't transformed the data, we're just reading the raw data. Oh, here we have it. Okay, good. We have a plot, right? So this is the one that I was showing. So especially these data, right? This you're going to have it duplicated by a layer. So we have the RNA layer for now. When we transform the data, we have another layer which is going to call SCT, and it's going to have also these tables in SCT. And they represent slightly different things because the data is being transformed. So the words are layer, assay for RNA and SCT, and layers for this.
So what is the scale counts, raw counts, then the normalized count?
We still don't have it normalized counts in our data because we haven't normalized anything yet. So currently our Seurat object has only the layer for the raw counts. It has assay, RNA, layer, raw counts. Yep.
After undergo processing, then we will finally analyze this.
We're going to be filling the Seurat object, right? Initially it's a small one because we just have the raw counts. So, and again, everything I'm saying verbally is explained here, and I always say whatever I say here. And on the text over, it's better than whatever I'm saying, I think. Overrules whatever I'm saying. That's my guess. Glimpse is part of the, I think, the tidyverse, and it gives you a little bit more details about the object. I don't know if we need to go into details. But just telling you everything is inside of that object, right? You can see that we're going to have counts, barcodes, and this gets even more complicated when we keep adding assays. One important section of the Seurat object is the metadata. Similarly, the metadata we have for bulk RNA-seq, there is a metadata table inside of the Seurat object. So, you know how to visualize it, right? We talk about visualizing, visualizing, and visualizing. So we can do, for example, to access it, we use this add symbol, and we say, oh, go to the metadata table, and you can go to different tables inside of the Seurat object, but we want the metadata, metadata, and we're going to print the 6 first rows. So what do we have here? So what is this? Which one, barcode or UMI? Barcode. This is.
So is the metadata table we are accessing the cell metadata or the gene metadata?
You're accessing the whole sample metadata. So these metadata is so these are cells. This is the ID for every cell. So this is huge, right? This needs to have 9,000 rows. So we give them the original identity is given by how we name the project. But if you have multiple samples, you can name them differently, right? So this, we use it as original identity, but you can have multiple samples. Later on, we can change this to be a cluster. We can change it to be a cell type, whatever we want. And we can keep adding columns to our metadata, like clusters, cell typing, whatever you want. We can add a lot of, and we'll see how we add it.
So these are indicating different samples of the same type. So these barcodes.
Yep.
Different cells.
Cells within the same sample. We only have one.
Only one. Okay.
We only have one sample, and these are the cells. And we name our sample, it took the name from whoever we called that project from. But you can have a single Seurat object with multiple samples in it, if you want to. I mean, if you have a way to identify them. So, so the number of counts, so this is our counts currently, right? And the number of genes that we have on the sample. I know I'm doing a bunch of things now just to show you how we can add more metadata into our column. There's already functions inside of the Seurat that allows you to do that. It's quite common that we see a lot of ribosomal protein genes being expressed in our data, and sometimes you want to remove them. You want to do what we call downplay their role in our analysis. So we can calculate how many we see in our sample. So here what we're just doing is trying to get all the gene names that are ribosomal proteins. Not ribosomal RNA, proteins, okay? Genes that are part of the complex of transcription. So here I'm just testing, out of the genes that I detected, how many are going to be ribosomal proteins. That's the only thing I'm doing, just to do a quick test. And it detected all of those. Those are ribosomal proteins. And the only thing, I mean, the problem is if it's busy doing transcription, you're going to have a lot of these being expressed. And I don't know, maybe they are not important for your biology, and you want to downplay them or remove them from the clustering process, or do something with them in the sense that you don't care. It's one of those noises we talked earlier. You don't care if the cell is doing a lot of transcription and is just grouping together cells that are doing transcription. You want to group together cells that are T cells, not that doing transcription, right? So this is biological signal that you don't care. And it's quite common sometimes that you see a lot of expression of them. That's the only time, I mean, I'm putting it here. It may not be a problem that you care. Also, like division, cell may be dividing, and you don't care, you don't want to group cells that are dividing because they're doing a specific process. You want to group cells that are T cells. Again, except if you're studying cells that are dividing. That is a different story. But there are ways to kind of filter this type of data.
They will always come to the transcription data that we are analyzing. Be
Yeah.
it bulk or be it single cell RNA.
Yeah, yes, the advantage of bulk is everything gets diluted, kind of thing, right? Because you're doing the bulk thing. All these lines here, the only thing I'm doing is calculating, for example, what is the percentage of mitochondrial genes. So I'm taking every gene that starts with MT dash, those are mitochondrial genes. So I'm telling Seurat, calculate the percentage of this gene and store it in the metadata called column %MT. Now that I know that all these are ribosomal proteins, I give it a way to select the ribosomal protein genes, and I'm asking, oh, can you give me the percentage of ribosomal that you have in that sample? And then I decide, okay, what if I care? I don't know, you may care about a specific gene. What is the percentage per cell of that gene being, the percentage of transcripts on that cell that belong to that gene, and give me the percentage, right? And all this, it goes to the Seurat object and is stored in the metadata with these column names. So if you run the two next, right, if we explore again, let's be sure, right, let's calculate this and be sure that it's been stored properly. You see now the metadata is much bigger. So this is our first cell. And now you have, this cell has 5% of mitochondrial expression, right, or transcripts, which is pretty big if you think about it, right? So only 16 genes, no, I don't think there are 16, but some few genes are already taking 5% of your data, the mitochondrial genes. You can see these other genes are in lower percentages, right? But look at the ribosomal.
9 to 16 genes they are in total. They are encoded by mitochondrial DNA.
Yeah, 16, right?
I think 9 or 16.
I don't know.
I'm not sure, but they might not be affecting the data so much because they're too low as compared to the whole genome.
Yeah, but it's 5%. Or what is it? In this case, it's 5%. The whole expression in this cell is already only 16 genes. Right? And you're going to see that you're going to have some cells that have 10 or 20. For now, we're just adding this information that we may use later, right? And you can add other information that you want here. But it's added by a cell level, right? So we're adding all this information at the cell level. You remember this. Well, I explained it here. This is a way to just, the display window, I'm adjusting the size so I can see the plots better. So the next line, now we can start doing visualizations. So we can only see 6 lines here. I don't get a global picture. So let's visualize the percentage of ribosomal RNA, the percentage of ribosomal proteins, PCAM1, and this other gene. And we can do the same thing for the number of features, the number of counts, and the percentage mitochondrial. And we're asking, okay, what layer do you want me to plot? You can be explicit. Give me counts. I'm plotting counts. We still don't have the normalized counts, but you could be explicit of what you want to, what layer are you plotting. In this case, it's just the normalized counts.
So here we can define raw.
Forget about the warnings. Oh, by the way, we're using, as I mentioned probably a couple of times, the single-cell RNA-seq field evolves really fast. Software evolves really fast too. We are constantly updating packages. Well, you don't have to constantly update it, but we do sometimes. And then we encounter that some CRAD is doing their best to always be compatible with other packages that also get updated in their own, like ggplot, and there are incompatibilities, right? And sometimes we get some warnings, especially this is the latest CRAD, which is 5.3. There's a big jump between CRAD 4 and 5. They change a lot of things. You need to adapt. And in 5.3, there's a lot of issues with other packages incompatibilities. And sometimes you get warnings. So this is, I guess I did too much. You can see those plots, right? You're running your notebook in your own, so I don't have to probably show it the complete plot on the screen. So these are the BioLin plots. They're also density plots. You can see that every dot is a cell, and the area here is telling you, it's trying to define how dense is that fraction of the plot. What we plot, the first one was ribosomal proteins, right? Look, there are cells that 40% are just ribosomal proteins. They're taking a lot of sequencing. These were 2 different genes, right? In those cases, yeah, it's normal that you see very low expression, but we're just taking 1 gene.
What is this on the scale y-axis? It's the percentage?
The percentage, yes.
So in RNA sequences, we do not do rRNA.
Depletion, yeah, you do. You do in the library. They use polyase. They can use, I don't know what other systems they can use too, but Bonnie knows all the library preparation techniques. This is not, okay, just may clarify again. This is not RNA, rRNA. This is ribosomal proteins. These are proteins that are part of the structure that do transcription. You know, when you do transcription, you need proteins and rRNA to do the transcription. So this is the protein part of it. This is a gene that encodes for the protein. So if you plot a violent plot for number of features, or the number of genes and the percentage of mitochondria, which is the third one, right? So we were saying that the first cell, it had 5%, so it must be one of the cells that are around here, right? Maybe around here.
Can you just help? I have no idea what is happening here.
Sure.
Load the library and library can show you here.
So hold on a second. Let me finish this and I go. Any problem plotting this?
What is the difference between this feature RNA and count RNA plot here?
Yes.
What is this difference?
So one are the genes and the other are Sorry. Yes. So one are the transcripts. How many? The count of the transcripts.
Will be more, right?
Yes. And number of features is genes.
So the count RNA could be 75,000 on the scale. They are transcripts. So second is transcripts, right? Count RNA.
Yeah, I don't see the plot. I don't know how I plot it. But whatever you have in front, you know better than it. Yes.
Feature, what is the count?
Feature RNA.
That's number of genes. This is number of transcripts. This is percentage of mitochondria. But you can see this cell here, what it is. 80% of all the transcription is mitochondria.
Mitochondrial gene.
Yes. So what we said, this cell probably is dying, or I'm assuming it's dying. I don't know. I'm not sure about the biology, right? But these ones may have a lot of stress or are about to die. So maybe they are not interesting for you, and we could go and filter these out. Although they look a lot of dots to you, you have 10,000 cells. So don't be afraid to filter if they are not, you have enough cells for your experiment.
So this is in the plot. We are looking at the biggest area means this is the mean.
That's a density. So the red thing that you have below is how dense of dots that area is. Because if you plot the dots only, it's hard to tell how dense it is. I mean, you see that a lot of dots, but you don't know, you need something else helps to have. So they're plotting exactly the same thing. One is the density plot, the other one is just the dots.
So here we can interpret this PBMCs have approximately 2,000 genes and having between these transcripts between around 10,000. From this data we can interpret like this.
I don't know what they have approximately 2,000 genes, you're saying? On average? Is that what you're trying to estimate?
Yes.
You have that in the...
2162 genes they were showing, right? In 10,000 cells, the approximate gene was 2,000.
I don't remember. You said 2,000 on average? Okay, yes. So if the average was 2,000, it's 2,000. The average is really hard to read here, but maybe you can. So, any issues running things so far? So, plot the rest of things that are coming. There are 2-3 plots. You can plot them. You are visualizing the same data, but in a scatter plot way. I think the next ones.
First one, I'm not going to getting like it's saying syntax error. But why should there be a syntax error in here?
And you didn't change anything, right?
Yeah, that is the one step. Then there should not be any error. I don't know.
And okay, the problem is this is an issue with R. This is useless. The errors they give is quite useless because it looks like the error is here. It's not true. The error may come from earlier, and I don't see it. Let me start. I'm going to start removing lines that are not important, just to be sure that I don't miss anything. Okay, the error still exists. So one thing we do sometimes, this is not necessarily, we don't need that. Let me double check this. Okay. Oh, okay, I see the problem. I see.
I'm not in my notebook.
Nope. You are in the notebook for whatever reason. This
This is different kernel we have to use.
we used to use the R kernel,
Okay, it was using Python
the
kernel.
Python kernel. Sorry. Any problems reading the next plot? We are plotting the number of transcripts versus the percentage of mitochondria, right? So most of the cells that have highly mitochondrial expression at the same time have very low transcripts too. So
So basically, as the count of RNA increases, I guess the percentage of mitochondrial expression increases, right?
well, no, you would see a line. What you're saying is you would see some line growing here.
Yeah.
This don't, all of these mitochondrial of these cells with mitochondrial expression have very low count RNAs, so they are not expressing other genes either much, right?
So this is the total.
This is the healthy population, right? What we see here.
On x-axis, it is the total number of genes, total number of transcripts on
Yep.
x-axis.
On x-axis transcripts, mitochondrial in the y-axis.
Gene is mitochondrial gene out of total number of transcripts.
Yes, and which is low, right? So most of these cells that have high mitochondrial have low. You don't have cells here that has high mitochondrial and high counts. You don't have that.
Low transcripts of other genes.
Yep. So this one is transcript versus genes. Okay, here. I think this is probably a little bit more important too. We generally expect that the more transcripts you have, the more genes you detect. So you should see some growing line here. A linear, kind of linear correlation, right? We're going to be filtering this top section here. Generally, we say that if you're going to have doublets, are going to be sitting here when you lose this curve. You're sequencing more transcripts, but you're not seeing more genes, so this gets flattened. And we generally say that most likely this is caused by being a doublet. That's what we generally say. That's it's not necessarily true. We don't know, right? We don't have a way to test it properly. Also, if you remove this, oh, by the way, so here we already remove when we pre-filter, right? So these are when they don't have enough transcripts and genes, we remove this section here. You don't see cells with very low number of features or genes because we remove them, or transcripts. So, but this top section here, we generally filter them out and we say, okay, we are trying to filter the doublets out because we're losing that linearity and it's starting to have a curve. So you're sequencing more, but you don't see more genes. I don't know. If I tell you the truth, my feeling is we probably should use better algorithms to estimate the doublets, and you have them. So if you Google, there's one called SC Double Finder, and I remember there are others you can use to try to detect doublets instead of trying to remove arbitrarily with some cutoff. But it's fine too. Again, you have a lot of cells. Another thing you're helping is to reduce the spread of your counts, so the normalization also gets a bit narrower. So you make their life easier. And you can even cut this section here, right? These are cells that don't have much expression and don't have many genes. I don't know if they're important for your analysis. But they maybe are, I don't know. But you can be even more stringent and keep cutting and keep narrowing your curve of expression. So if you want to see how many dimensions, how many cells you have, this is one way. I'm giving some ways to access the data. I'm not saying anything special. So if you want to know, for example, how many cells we have, the dimensions of our Seurat object, we have 22,000 genes detected in total in our total cells and 9,000 cells. So we can do a test, or we can filter even more and then recalculate and see how many we filter. We can filter, for example, only keep. If you remember, we selected the cells that have 500. We increased it to 600.
Suppose we have a lot of doublets in our plot.
Yep.
Can we remove those doublets?
Yes. I mean, we are assuming they're doublets. That's, I don't know. It's a manual way to remove doublets, right? I mean, we don't have any tests to actually test if they're doublets. There are algorithms. That's what I was saying. You can use packages to estimate the number of doublets if you want to. So let's put it differently. It's one of those things that you can do an analysis, and if you see weird clusters, or if you see weird data, then you can say, oh, maybe it's caused by the doublets. Let me go back and remove more, or so on. So the iteration process we were talking about. So what we're doing here is subsetting our object. So we are filtering even more. So we are filtering the lower part of genes, right? We're saying we want to have, instead of 500, as we said originally, let's filter a little bit more and say 600. And this one is what I said earlier, right? I'm creating a cutoff line around in the middle here, right? Everything, all these cells are going to be gone if I use that. Why? Because I'm assuming that those could be doublets, and I don't want them out. So if we do this and check the dimensions again, so did we remove 4%, which is the expected, or 8%, depending on the, I don't know, the chemistry was used here? I doubt it, right? We didn't even reach 1% maybe. Can anyone do the quick math? How many we remove?
300.
We remove 300 out of 10,000. That's what, a 0.3? 0.3, right? So it's not even the 4%. So we didn't remove the expected number of doublets. We probably still have some there. Is it going to change the results? Maybe not.
Shouldn't that be 3%? Like if we remove 300 out of 10,000.
It is 3%? I don't know. I'm doing the math in my head.
Yeah, it's 3%.
Oh, it's 3%? Okay, then we remove close to 4. Okay. So, yes, you're right. So, yep. Just hold on. So we also mentioned that we can create a cutoff, right, about mitochondria. So that's arbitrary again. You can make the decision. Here I decided 20%. We can go back to 10% if you want to. Anyway, all those mitochondrial sections, I mean, you can be more stringent. Pick a number. Look at the plot that you have up there. You're going to see that most of the cells expressing high mitochondrial are all also not having many genes being expressed. So I don't know if that's important. So you can be more stringent than I did here. So I picked 20%. So what would remove? 300 more. So we can be more stringent. Yeah, we can do 10% maybe.
So if you look at this plot, mitochondrial percent only, most of the cells are expressing approximately 7% mitochondria.
So probably you shouldn't do less than 7%, right? Wait.
Maybe 10% or
Yeah, go ahead, remove it. That sounds good. Change that to 10 if you want to. But from now on, we may have different plots. Okay, they're suggesting that we should go down to 10. If you want to go down to 10, we can do that. It's up to you. But the plots from now on, if we have different numbers, they'll be slightly different. So you know how to do the next exercise, right? And how to save the plot into a PDF?
cells with percent mtDNA, that's not coming down, but they are changing these values. So when I'm putting it 15%, it is also saying the same number.
You just add 15% to this text. You are not filtering anything. Oh, you did. So you did. But it changed. Mine doesn't say 900 something,
So I'll
right?
change it to
Mine says 93.
So I'll change it, say, to 25 at both places.
No, it's not going to change. You already filtered it once. You cannot go up once you filter it one time.
Okay.
You remove them because you.
So maybe I can decrease it, but I cannot increase it.
Correct. Well, I mean, you could save it in a different object and then it would be fine. But we overwritten the object. So any problems with the exercise? I'm assuming no, right?
No, like I have one problem. So I tried setting like, I tried setting the filter really low, like for example, percentage with at 1%, and it's sort of crashed at me. So at that point, like I got a table with 6 entries, and then I tried adjusting it back to 5%.
You cannot because sorry, don't play too much. If you keep filtering down, because you have this object, you reward the object, and you don't have those cells anymore.
Put it back in.
Yes, to put it back in, you're gonna go up and rerun everything. Sorry, I didn't. I mean, if you want to be safe, you could have created another object, and then. But it's a bit dangerous to keep creating objects because they are big memory hoggers. I don't know, maybe, by the way, this is probably cleaner. No problems with the exercise, right? You already know that generally I give the answer somewhere. You just copy paste the code and modify it slightly. Do I need to explain? Basically, we are creating a plot with x and y, and we store it into a new variable. So we're storing the plots here, and we are plotting them. CRAT, when it plots, it uses ggplot2. I keep forgetting. I think there's a library called Patchwork. I don't know. There's an R expert here that likes to plot and knows. This plus symbol allows us to plot.
It basically like fuses them together so you
Yep.
can get them in.
Yeah. Yep. And I think the library that allows to do that is called Patchwork. So if we do a vertical line, I think it plays the. Oh, we're not going to do here. Sorry. My bad. Doing next to each other? Yes. So I know yours is going to be on top of each other, but if you do, for example, a vertical line, you can get it next to each other. That's the filtering that I use, 20%, right? So I don't have any more of those cells. I decided to cut off a 5,000, so I don't have that section over here. Every data set is going to be different, right? So the 5,000 cutoff that we use here doesn't mean it's going to work for you. So normalization. This is the basic normalization that CRAT does. As I mentioned earlier, they recommend to use SCT. That's the latest recommendation. So you could skip all this if you want to. We don't have to. Let's do it. And they're going to be stored in the same object, but in different assays, right? The assay of RNA and the assay for the SCT transform that we're going to do later. So you are not overwriting anything if you do this. So they use a log transformation and a scaling factor. So it's dividing. So the units are counts every 10,000 log transform kind of thing. We can identify the top features. The genes are most variable because they're, in theory, the ones that have more information, and the ones we're going to use for clustering or the ones we're going to be used for UMAPs. So we don't have to use the whole set. In that way, we are removing also possible noise. The genes that variable less, they could be technical noise and other things. We are focusing the ones that variable the most. There's no right or wrong answer about how many you want to take. I use 3,000, I think, to keep the same number of genes that SC transform uses as default, I think, later on. VST, variant stabilization, right? That name sounds familiar to you. We use it also as a bulk RNA-seq to transform our data. But in bulk RNA-seq, we never use this transformation for analysis. Don't worry about the warnings either. So basically here, we are plotting all the genes, all the cells, sorry, genes, sorry. In this case,
First, we have normalized the data, right? In the basic normalizing and scaling. Then now we are identifying the highly variable features.
yeah, correct.
So highly variable features means
The genes that they are more variable in our data set. We are assuming that those are the ones that contain more information. And we're just plotting them here.
Variation only over different cells.
Yes.
In different cells.
All over, yeah, all our data set.
Giving us the maximum information.
Yeah. So this is the same plot. Just plot label with the top 10. How many we said? The top 10. You see what I did here with head? Now I'm giving a number. I don't, by default, head only uses 6. Now I'm giving, actually, again, I could have done using the pipes, right? It would have, I'm not consistent in my way of writing code, but if I would have been more consistent, I could use the pipe and say head 10, and I'm getting the top 10 genes. If I change this number, I could get the top 20. Now what I'm doing is just labeling the top 10 genes. They're most variable. I don't know, I'm not familiar with them. I don't know what they do. Again, similarly we did bulk, we're just exploring the data. We're not doing any specific analysis yet. So if you, let's see.
Variably transcribed across various cells.
If you print again the object, now we gain another layer, the data layer. That's the normalized data. Still, we only have one assay, which is the RNA one. So we still have another layer that we can fill, which is the scale data layer. And similarly, I tried to explain that in the heat maps when we scale our data by row. So this is the similar approach, the same thing. I'm blocking the view, people. I don't know. I don't know if I should standing up or not. Is it wanna be one of those steps? No. Okay, it's done, right? So now 3 layers. So we have our the row counts, normalized counts, and the scale data. We're going to use the scale data for the next steps. Again, any questions? I probably give more details in the text than what I'm talking. And as I mentioned earlier, you probably can skip all this and go to the other type of normalization, and you do it in one step. So you see we are running a PCA too for single cell. Again, the PCA is only to keep reducing our data to a more manageable number of dimensions. So when we do clustering and UMAP and TSNES, we don't have to deal with the whole data set. And we are only using 3,000 genes. It's the one that we selected as the top variable genes. We are not using the whole data set because, again, the PCA generally don't care about the genes that don't have any, they are not they don't vary. Thank you. Yeah.
So from 22,000 features, the 3,000 features were highly variable.
Yes.
And here we have represented the 10 highly variable.
Yeah, we just label them just to know where they are. You may be curious.
Now we are using these 3,000 for plotting for the PCA.
For the PCA. We are not going to plot the PCA because it's not going to give you much information. Again, the PCA is a linear process, right? And it's not going to represent the data that well. That's why we use UMAP. But again, pre-step UMAP or TSNES, you even reduce the dimensions more with the PCA to be everything more manageable. Did it finish for anybody? I don't know. No? Okay, let's wait. We have enough computer power today for sure to run this. So. Questions so far? Again, take these moments of waiting. If you want, you can reread whatever I wrote there. There's more details. I always forget details, so it's better if you read the text. Oh, it's on? Okay, good. No, I was just checking. It seems that it's something that I don't know. Internally, maybe is parallelizing the process. So meaning that if it can, it's going to divide in multiple jobs. So it means even if our cluster has 100 CPUs, some people of us may use multiples, not just one. So we may encounter, I guess not like yesterday, but it may be slow for some people. You got it too? Okay, so people are, it's finishing. Okay, good, good. So plot the next thing, the elbow plot. Yeah, the elbow plot is the same as or similar to the scree plot. Remember yesterday were the bar plots where we're saying PC1 expressed so much and so on. Sorry, PC1 explains certain percentages of variability, then we have another, and it goes down. Remember that scree plot? Yeah. So this is similar. Yeah. So it's doing the same thing. Oh, mine finished. We generally don't make use of this that much, but it gives you information about, again, the main contribution of genes for every PC. It separates by the level of contribution, I guess. The elbow plot. This is similar to the when we evaluated the percentage of variance explained by every principal component in bulk. We created that bar plot. It's the same thing. Right? Why are we plotting this? When we use this type of normalization, you're going to make a decision, okay, how many principal components I'm going to use downstream. Right? A way to do it is using the elbow approximation, they call it. So when you see that the dots flatten, right? When you let's say the elbow, this plot is going to indicate that you should stop using PCs. So that is the number of principal components you're going to be used downstream analysis, because everything else you're not gaining much information. It's also assumed that these principal components may contain noise or data variants that are not important for you. So any suggestion where we should stop taking principal components? Around here, maybe 6. Yeah, that could be a good one. We can maybe go up to here, maybe 10, right? Again, we are not going to use this normalization for anything, but I just wanted to let you know that it exists.
I just wanted to understand what is the concept of this elbow plot. Why do we need to, how do we interpret this?
So I don't know if you remember yesterday, the principal component analysis, what it does is to create new axes of information, which are linear combination of genes, blah, 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 and you get them here, right, those genes. And then, so principal component 1 is the principal component that explains the most variable of the, or the most the standard variation in your data, or the standard deviation of your data. That's the one that explains the most. And the other are sorted, right? So instead of using all the principal components for downstream analysis, we're going to make a selection of only a few of them. The idea of the elbow plot, there's other plots you can use to select. Decide to select some of the principal components. Again, I don't think you may use this approximation. I'm just showing it. And one of them is using the elbow idea of saying, okay, once I see that if I keep adding principal components, I'm not adding much information, you could stop there. There's the assumption that these probably are just noise and not signal. Yeah, so that would be a way. So if we keep doing now, so if after this we decide to do UMaps with this data, you could say only use the 15 top principal components when you build a UMap. Only use this to do the clustering. The problem, of course, it could be that, let's say, I don't know, you care about T cells that express CD34. And CD34 is important for that principal component analysis. You're throwing it away, right? It may not be ideal. There's way you could explore this and be sure that you're not throwing away important genes for you. And there are other ways you can access that data. This type of normalization may be useful in some occasions, and it was the original one for CRAT. But nowadays we use this other one, the SC transform. So you can run this. We can start running this because they may take also 5 minutes to run.
So what is the positive and negative?
That's, I don't remember what is positive and negative here.
One is down-regulated or genes
That's what we have done. Yeah, but you're not comparing anything yet. Yes. So there's no way to know if it's unregulated or not.
So does it mean the PC1 is including these genes?
Yes, yeah, those are the ones that have the most loading. I don't know if a negative loading makes sense here. I'm not sure. Yes, yeah. So we're changing type of normalization. So now we're going to have an assay with the RNA normalization we just done. Now we're going to create a new assay that is the SC transform normalization. When you are adding a new assay with 3 more layers. So you are correct, as I said. Yeah. So in here, we are asking in this transformation, by the way, there are different methods. I picked this one. Probably I read at some point that this is the one that recommended, and I stick with it. There are other ways to do the SC transformation. I've never done a proper analysis to compare them. There may be papers out. Again, it's one of those things that you could iterate if you want to, right? And see whatever fits your data better. I'm asking with this variables to regress. Percentage of mitochondrial and percentage of ribosomal RNAs, proteins expression. I'm asking to SC transform to downplay their role, right?
Or to delete them altogether?
No, we don't delete them. We ask them to downplay their role.
What do you mean by that?
Now I tell you. So I'm going to, with an analogy. No, I'm not explaining exactly how it happens, but with an analogy. So when we normalize, the first thought you have about normalization is saying, let me divide the number of counts by the total number of reads. Basic normalization, right? It is already happening. I don't have to tell him. You need to downplay the role of number of reads. That's what I'm trying to say. So we could add here number of counts. But it's already happening, so I don't add it. But on top of downplaying how the number of counts in the normalization, so what I'm trying to tell you, if a cell has a lot of counts, they need to be normalized, right? Because if you're comparing one that has fewer, you're going to detect less, but it has fewer reads, not necessarily fewer expression, right? So you can normalize those things. So you can also then play the role of ribosomal proteins. You can then play the role of mitochondrial. So I'm trying to then play, or I'm going to call it be part of the normalization process. So I don't want ribosomal RNA being a factor, an important factor in my analysis. Can you remove them? You could, but I don't know if it's important for us later on those genes or not. So I don't know. I get them. So for example, for the UMAP, actually I've done a recent analysis where it was with T cells. They already knew what T cells they were working with. They didn't care about that. And the clustering was just clustering group of T cells, subtypes, and all that, and they didn't care about that. They wanted to see how the drug they were using, it was affecting the T cells. And they wanted to see some separation of different subtypes. So what we ended up doing is, for clustering only, we decided to remove certain T cell markers who were driving the main clustering. So can you remove ribosomal proteins from your clustering process? You could. For now, we are not removing anything. We're doing a standard analysis. So this is what I was trying to say. So now we have two assays with 3 layers, each one of them. So the current, does it say the current one? Yes. Active assay now is SCT. So from now on, if for whatever reason we want to access the other assay, we've been doing the normalization process, you need to be explicit, because the only one that is active is this one here, the SCT transformation. If for whatever reason we want to see the RNA normalized counts, I don't know, for whatever reason we want to plot it, we need to access that data explicitly. It may be that you load some package, R package, that doesn't use what SCT transformation creates, and it uses the expression data, so then you need to access that assay. You need to be explicit. For now, we're going to leave it active because that works for us. So again, if you want to do the glimpse thing, you can do it. The metadata didn't change. We have a new assay. We need to run the PCA again. Sorry, guys. Because now we are running in the SCT transform data, not the other one. So I'm asking to plot again the elbow plot, and it should be slightly different than the previous ones because, again, we are not using the same data. And probably this is going to be slower. After this, everything gets a bit faster. I hope so. So, sorry, what? Yeah, the active assay now is SCT, is not the previous one. If you're curious how much memory we're using, currently we're at 10 gigabytes, 11 gigabytes of RAM. So you can get an idea if you can run these on your computer or not. Probably we could save memory if we wouldn't have run the normalization process earlier. It's on? Okay, good, good, good. So it's running faster than previously?
And here is the estimated distribution between two of the.
Oh, I will plots, yeah. It's on? Okay, good. Did it finish for everybody? No? I'm just going to keep going. So you have it on the screen now, the new elbow plot. It looks slightly different. If we needed to create a cutoff now, it's even harder. I don't know. Maybe here, maybe here. So at the end, it doesn't matter much because we are not going to make a decision based on the elbow plot. For the SC transformation, they don't recommend anything. They tell you get enough PCs to be sure that you get enough information and you don't get it enough. It's a balance between noise and signal. So you need to make a decision. If you go to the vignette in the website, I think they take 30 principal components. So you can take that. You can run your UMAP and see if you get the nice clustering and do the clustering and the UMAP and see if you get the expected results. And play with those numbers if you want to. So I was just showing the elbow plot, but again, for SC transformation, you're not going to use it. So, but at this point, we need to make a decision or leave the defaults, if that works for you, about when we're running the UMAP, how many we're going to use, this process of finding neighbors and finding clusters. So how many we're going to be using? In this case, I choose from 1 to 30, so from the first one to the 30th. You can try 50 and see what happens. If you try 100, it may be too noisy, the data. I don't know. Principal components. So from the first one to the 30th, you can change and decide not to use the first one because maybe the first one is full of ribosomal protein genes and you don't care about that. So then you can say, I'm not going to use that principal component. I'm going to use the rest. So I'm assuming this is going to be a bit slow, maybe? Those are the most computer-intensive steps we're doing now. Oh, it's faster than I thought. Are you guys here? Well, I should probably tell the plotting. I don't know. Should I explain it? So I'm assuming everybody knows what we're doing here, right? We said we create a PDF device because we want to store whatever we're going to plot next, and we close it here. So we are plotting this dim plot. The dim plot is how we plot the UMAPs. I'm asking, okay, take the object, do a dim plot. We already calculated. All the information is done here, right? Run UMAP, it's already calculated the coordinates of every cell, so it's done earlier. So the only thing we're doing with the dim plot is accessing that information.
So what is there in dimension 1 is to 30?
The PCs.
PCs.
Principal components we were using.
That we can define ourselves.
Yeah, and we're saying from the first one to the 30th one, but the first number you could even change it and say from the second to the 30th and remove the first principal component. I'm not sure why you want to do that, but it could be that the first principal component, all the genes, for example, are ribosomal protein genes. And you know that those are there, but because they're doing transcription, and the transcription process is something that you don't care in your biology, and you could remove them. I'm trying to create an excuse to do it, but I don't know if that happens often or not.
Why it's in minus 10?
That's coordinates. It doesn't mean anything. Oh yes, I was explaining the dim plot, right? We're talking about the dim plot because we already done the clustering. Oh, I should probably, let's do one thing. So if we explore, how would you show the first 6 lines of the metadata table? With head, right? So just do it. So we're accessing only the metadata table inside the Seurat object, and we want to show the first 6 lines of that. We did it earlier, right? So
Now we have to rerun the GIAN to get this metadata.
hold on. UMAP, we already done it. So the metadata is not related to the UMAP. There's no UMAP information in the metadata. But we run the clustering, right? We ask run cluster, and we give them all the information and run cluster. The clustering, what it did is add new columns in our metadata. So every cell now belongs to a cluster. Every cell now has a new identity. The cell stopped having the original identity we call PBMC something. Now the active identity changed to be Seurat underscore cluster, which is this information here. So the first cell that we have in our metadata belongs to cluster 0. These two are identical, and I'll go about that later. But if you, I don't think, probably if you do glimpse, I'm not sure if it's express. So glimpse, Seurat, let's see. Yes. Let's see if it's also without this. No, it doesn't say. Okay. So we notice that we run the clustering process, the UMAP. Now we have a new column in our data because we run the clustering, which indicates the identity of every cell for a cluster. It assigns a cluster to every cell. That's the clustering process, right? But now there's another thing that changed in our Seurat object, which is the identity of the cell. Stop being the first column in our metadata was the original identity, right? So if we go back, every cell was labeled as original identity. So human PBMC. Now we have a new column called Seurat cluster. Because we run a Seurat cluster automatically, let's see if I found that line. Active identity is the clusters. So it changed. So it's something that you need to have in mind. You can even create your own identities and add it to the metadata and say from now on the active identity is not Seurat cluster, it's whatever I say it is. So you could do that, and we're going to do it later. So we can say, no, cell 1 is a T cell, so his identity is T cell.
We can give identity like person or like per clusters.
We're going to do it per clusters, but it's going to be, at the end, is a group of cells. So what you're going to see in the metadata is by cell. But the way we do it is better than just labeling cell by cell. So that's the reason when we come here and say, plot the UMAP, plot the labels now are the Seurat cluster labels, not the original identity. This is just to make it more readable, like a box around the number because it's hard to read. So now here it is. We have clusters, a UMAP, but we don't know exactly where they are, right?
In a given cluster, can you see which are the genes which are there on the basis of what genes they are to be clustered?
Yes. Yes, we can do that. That's we're going to do later. So we can figure out why cluster 15 that must be behind the label box. What makes cluster 15 cluster 15? What genes making B cluster 15? So for the sake of time, I'm not sure that we should do the next exercise. But here I give you instructions of all the parameters that you can change. If you have time at home or whenever you have Jupyter notebooks available or time, you can play with all these parameters if you want to. I create this table so you can see what happens when you increase a parameter. So we talk about this, right? We can give it more than 30. We can give it 50 and 60. And again, this is general expectations. It doesn't mean that it's going to happen to you. So you get the effect when you increase it. So it may include noise. So something it may change in the UMAP. You may miss information if you include less. This is what we're talking. So you have more parameters that you can play with for clustering or for the UMAP and so on. I don't know if you noticed, but in the metadata, the last two columns, there was one that says clusters underscore 0.8 or something like that, right? That is because we use this resolution parameter. This is the default one. We could change that, and it's going to add another column with a different resolution parameter. I like to play with this number more than the others, but I mean, you can play with all of them. But we are not going to do that. I think it would take too much time if we export all that. So I prefer we try more things this afternoon than playing with this. So don't do the next exercise. There's a way to explore several values. And I don't think you may not need to do that for your analysis. But that resolution parameter for clustering, you can tweak it, right? So a way to explore several values is using this approach, which is called, we're going to give it instead of a single resolution to find clusters, we're going to give them a range of resolutions. So it's going to be from 0.2 to 0.4 in increments of 2. So instead of testing one resolution, we're going to at once as to find cluster to try several of them. So that implies that our metadata is going to have all of a sudden, I don't know how many, 7 more columns, 8 more columns for each one of the resolutions. Each one is going to have a different cluster, right? Oh, actually, yeah, the next step is exploring the metadata again. So this is quite common in a lot of functions that print a lot of things on the screen. I'm telling, don't explain me what you do. I know what you do. Don't show it. This is becoming quite big. Right? So now we're testing 0.2, 0.4, 0.6, 1, 1.2, 1.4. The 0.8, we already have it. So it's there. So it seems the first cell probably doesn't move from their clustering, it's stable. So now we have a lot of information, and there's a tool which is the next step that is called class tree, that what it's doing is taking every column that we just created and analyzing how stable a cluster is. So this is what it was, 0.2 resolution, the first one. So these are the number of clusters we found at 0.2. When you increase resolution, the first thing you notice is we increase the number of clusters that we find. So clustering doesn't mean it may not mean anything biologically relevant to you, because by default we just use 0.8, but what if we have used 1.2? We're going to encounter more or less clusters depending on the resolution we use. So if we want to see how stable is a cluster, meaning let's look at cluster 7 or the last column. It seems that it's stable all the way down. It doesn't change. So that population of cells that were classified 7 initially, with that exception that more cells are coming from 0, every other resolution, it seems that it keeps that group of cells always together. It's not changing. Cluster 0, right? It seems that it's not that stable. It keeps getting more divisions, depending on the resolution that you work on it. So we are working with the middle one, right? 0.8, I think. It must be this one here. So it seems that even if we increase a little bit the resolution, it doesn't change. It's stable. So you can see how stable is a cluster, how much subdivisions you can do with a cluster if you increase the resolution, for example.
What is defining resolution here? So if the cluster is dividing or getting into 2,
Yes.
or upon increasing resolution, right? So does it mean that the genes which we taken into account into one cluster, they were the part of 2 cell types?
So dividing cluster 0 in multiple subclusters, it doesn't mean there are subtypes or again, that's what I'm trying to say. First, the clustering doesn't need to match your expectation of the biology. So that's the message that I'm trying to convey. It is based on the expression, right? So it is not artificially creating clusters. It's just based on the expression. But it doesn't mean that if what you care is about cell types, it doesn't mean that every cluster is a cell type. It doesn't mean that. Another thing, cell types is also artificially created by us, right? Biology is not saying, oh, I'm a cell type, right? Of course, it has a meaning in biology. Okay, we create groups. We need to create identities to be able to work with them. That's an important message. So that's something that I always try to convey to people that look at our analysis. It's like, okay, what is your unit of work? Right? So if you're doing a single cell, what is your unit of work? Is a cell? Is a cell type? Is a group of cell types? What it is? What you're going to be working with? If you're working with T cells, then you're working with the unit of work is going to be some sort of T cell group of cells. That's your unit of work. If you want to go to the subtype level, then it's T cells that express CDD, whatever, I don't know, one of type. Then your unit of work is subtypes. Then we need to work with that resolution parameter to be sure we're capturing your unit of work. So that's what I'm trying to explain with the resolution. That is one of the parameters that you can use, but you can use others.
So whenever we perform single cell RNA-seq, our main purpose, whatever projects we try to put into that, so what are the cell types? What is the predominant cell types, suppose among T cells?
No, not necessarily.
So what is the cell type which is predominant?
If you work with single cell RNA-seq, the first thing you need to do is to write your question. What I'm trying to answer when I do single cell RNA-seq. If your question is to work with T cells, that's your question. Your unit of work is T cells. Your question needs to be related to T cells. There's maybe other people that they want to do some sort of discovery. They are not going to be able to define what cell type are working with, but they may need to identify that unit. They may work with only the cell types or T cells that have some expression. I don't know. There's different ways you can express the question and your unit of work. So there's not necessarily a pattern that you need to follow. There's people who sort cells, and then you already know what cell type they're working with, and they only care about if they're expressing some specific gene. They work with retina. I remember people working with some type of retina cells, and they do a knockout, and they want to know how specific cells react to the knockout. So then the question is different. But yeah, generally there's going to be some clustering, that's true, but not necessarily you're going to be defining cell types per se. But we're going to do it.
In the human, we have two conditions. So suppose upon treatment, before treatment, then what are the
That's quite common,
that are
yes.
changing. So if we go into high resolution, that will give us sometimes the rare type of population, or which may not be resolved on.
More resolution doesn't mean better data. You need to have that in mind. That's not true. Resolution, as I showed you earlier, it just creates more clusters. It's trying to subdivide even more things. But that doesn't mean that is biologically meaningful to you to have more clusters. So remember that I told you that there are ways to assign new identities, because now we run from 0.2 to 1.4. The last one identity we use is the 1.4 one. So I'm telling to Sirat, no, don't use that one. Let's go back to the original one. The original one is called SCTSN, that one, right? The resolution 0.8. That's the first one we calculated. With this identities, Sirat, I'm telling use this as the identity that I want to be active. Before I run this, so if I do the glimpse again, glimpse. Oh, I don't need to do that, right? So if I do write down Surat, what happens? Okay, no, it's not going to be helpful. It must be a function probably that I don't remember now to check what is the active. What is the active clustering? Does it tell me? Oh, active identity. It doesn't tell me what column it's using. But it must be a way that I'm not remembering now how to check it. Anyway, if you want to be explicit about all the resolutions we are using, you can do that. So if we want to change the column called Sirat clusters, we can do that too. I'm saying use the column, this column here in the metadata is going to be called Sirat cluster added to the metadata. That's what we're doing. We may have a way to create other type of identities, and we can add another column to the metadata using add metadata to the Sirat object. What metadata you want to add? What is the column name? That's what we're doing. This column already existed. What we're doing is just overriding it. So you can ignore that. So if I want, I can plot different things, right? So I'm asking use labels true, and the labels you want to use is the original identity. We only have one original identity, which is the project name. But you could. Let's imagine you have multiple samples. Then you can use this and plot the multiple samples, like the plot I show you on the slides. We only have one. That is pretty useless in our example. There is this other plot that you can overlay information from the table, from the metadata table, and ask him, okay, show me the number of, overlay the number of genes per cell, right, and give me a scale. So it seems that this group here is the one that we detected more genes. And I don't know, all of them go from 1,000 and up. I don't know, there's probably another group. I don't think this is helpful now, but I'm just showing the capabilities you can have, right? We can also plot, if you want to try, do I have exercises so you can try?
Yes.
Yeah. So overlay the expression, the mitochondrial expression. All these feature plot, dim plot, all are Seurat functions. So they're created by Seurat. And they use ggplot2 in the background. So you could, I think you can treat these as a ggplot2 and use the functions from ggplot2 to even modify it further. The only thing you need to do is to replace or use the column in the metadata that you want to plot. The column in the metadata that contains the percent mitochondrial is called like this, I think, right? So it worked. We don't have any specific cluster that it seems to be stressed or dying. If we wouldn't have removed the mitochondrial percentage, maybe we would find a cluster in somewhere that only express mitochondrial genes. It seems that there's a little bit of a high expression of mitochondrial here, maybe here. I don't know. This can be useful in some situations, maybe. You may, if you did you didn't filter too much in mitochondrial expression, maybe there are clusters that only cluster to, that you might put together cells just based on the mitochondrial expression. Questions? Am I going too slow, too fast? Let me know. So you can plot more than one thing at once, right? So in here I'm asking, okay, these are two columns. If you remember, at the very beginning we calculated the percentage of expression. Percentage of expression of this two genes in our cells. This is just I'm indicating how big I want the cell to be, and this is the number of columns that I want the data to be used. Again, some of these things, if you Google or Access with a question mark, you get a help about all the arguments that you can use for this. And again, this is a ggplot2. You can also ggplot arguments of functions to even modify it further. Okay, so CD, it's 30, 3D, okay. I don't know, I chose it because it was a marker for something. but it seems that it's concentrated down here, right? It's fairly specific for these cells. It's barely expressed anywhere else. These are the one, become one, it seems expressed here, okay?
So that would be expressed in certain group of cells.
Yep.
Endothelial cells or red blood cells, and this is C3D.
Yep.
Actually C3, CD3D.
T cells?
Um,
T cells.
T cells?
It's a I mean, like,
3D.
it's a co-receptor for T cells.
Okay.
So you have delta, you have epsilon, you have
That's what I thought it was. Here we have it. So maybe there are T cells.
Epsilon is
Okay. So what I'm asking, visualize the UMAP embedding color by cluster, and next plot a biolin. Okay, next to it, plot a biolin plot with the CD3D expression. So P1 is the first plot, P2 is going to be the second one, and you're going to be plotting them next to each other.
So we have to plot, right? Violin plots.
Biolin plot. I'm telling you what you need to do in every step. And we already done this. Maybe not with this gene, but we've done it with other things. Sorry, UMAPs, generally I call them embeddings. So they're stored inside of the Serac cluster is called embedding. When you call about it, so a UMAP plot or a UMAP embedding. So I may use, if you see embedding, that's what I mean. It's a UMAP. I could remove this and probably you understand what I meant better. Did you get anything good? No? So pretty much in every plot, every function, you always need to provide the object or the data. That's the first thing you do, right? And I think I mentioned generally when you give arguments, is always the name of the argument equal to something else. It's quite common in R. Sometimes we skip the name of the argument because some of them are always expected first, positionally. So that's why, but I think if probably the name of this argument is going to call object equal Seurat, you could do that and probably you get the same result. So this is what we did earlier, right? But we don't want to group by original identity. I don't know if we've done other plots. We want to group by the Seurat cluster. Cluster, plural. Okay, thank you. All right, that's the one thing we want to do. So I think we've done all these earlier, right? I'm convinced because I copied this. Yep. So, and what I'm asking is to use a Biolim plot, which is the ones we used all the way up. Yeah, a Biolim plot, right? So if you don't want to type everything, right? So it doesn't matter. Okay, I don't need to copy anything actually. So we first give the object. What would you want to plot, right? That's the question. How we call it? CD3. I think we call it like this, right? In our metadata. Yes? But there's something else missing. Well, we can plot it like this and see what happens. Did anybody reach at least this point or something similar? Yep. So if we try to plot this, it's not exactly what I'm asking. Oh, well, it works because you still have the identities. Oh, sorry, it looks very ugly now. Now, it looks a bit better. Okay. I mean, you can do really a lot of things with plots in Seurat without knowing too much about ggplot2. So this is the expression of CD3D that we said it was concentrated down here, right? That's what we observed in the last plot. Yes, so CD3D is at the very bottom except this group here. So cluster 1, all these clusters except cluster 8, they should have some level of CD3D expression. Again, the dots are not going to be that helpful. Maybe the density plot is going to be the most helpful thing. So these clusters should be here. Which is cluster 2 where? What is the cluster 2? Oh, it's up here. So cluster 2 is not expressing it. Cluster 0 is not expressing it. And of the group down here, the only one that doesn't express much, CD3D, is 8. 13 is expressing, oh, 13 is here. Okay, good. So 16 is here. Yep. You could be explicit also on the Biolim plot. I'm assuming Biolim plot takes by default whatever identity is the active one, but in some cases it may not be that Seurat cluster. This should be a valid way to do that too. You don't need the group by probably in the Biolim plot because it takes the active identity. All this code is somewhere up in the notebook. I didn't create anything especially new, except the only thing I asked to change was this, right? The feature. Oh wait, hold on. Oh yes, the dim plot without this is the feature one. That's the only thing I'd ask probably that is different from the rest. Oh, you see here I'm using the ggplot feature to add a title. So I can use ggplot calls to create titles. I don't know if they have a feature. Remember the tab. So if you want to, you don't know what parameters are available, if you press tab, it will show you in the function call what is available. All these are arguments that you can modify to use dimplot. But again, you can always modify it using ggplot calls. Questions about this? I'm just showing plot capabilities. I'm not trying to teach you how CRR works, just to tell you how I can explore the data. Right? We see how we can overlay gene information. We can also keep the UMAP next to some Biolim plots so we can see what clusters express a specific gene. If you have a lot of clusters, I don't think this is that useful though, because it gets harder to read. There are better plots for this. Yes, the identity is the cluster because we also, we can be explicit to say how you want to group the data with. Actually, the cluster is the active identity, so I'm assuming if I remove that, it would plot clusters too. So you don't. So if I remove this, it's also plotting clusters because it's the active identity. We can change the identity that is active at any time too. So these are columns in the metadata, right? Okay, can you do the next exercise? And probably you're going to be break for lunch. I'm giving you already half of the answer, right? So I'm talking to you by my eyes. That won't help.
So these to the notebook. First thing I want to remove all these things, whatever I executed, and save this to the notebook. What to do?
Oh, you went. Let me turn off. gene within that cluster. It could be that I have a highly What do you think? Your job it will be easier? So, yeah, any questions about dot plots? So we've seen Bioin plots, we've seen feature plots, the dim plots, and the dot plots. I think we cover pretty much all the basic plots in CRAD to explore the data. Except if I forgot any, I don't know, probably
And it was good that like everyone could do it on time. I was very skeptical whether it is going to work for everyone.
So one thing we could do before we leave for lunch is to run the next step, and then I'll explain it. Because this may take also, I don't know how long it's going to take, just in case.
Sir, can you show the how did you make
Command
that
line.
command you put dot plot to save, right?
Oh, what I was explaining. So remember our essay is SCT, is the one that is active. So what I was saying is they call it average expression, right? For me, it's hard to imagine. We transform so much the data that, yes, it's expression, but it's transformed. I would call it SCT transform average expression if you want to. I don't know how to call it. The units, it's hard to tell what unit we're using here. So you could, in a dot plot, you could specify, well, instead of SCT, use RNA, right? And then it would go to the other assay that we have and use that information for the dot plot. And these are all the options.
How did you get them? Like, how did you get
This menu?
no, the tab.
Oh, just, so let's imagine that, so currently it looks like this, right? This is what we run. I want to add a new parameter. So the cursor is now here. If I press tab, Jupyter Notebook is smart. It knows that you want to run dot plot, and it checks what is available for this function. Those are the arguments available for this function. I don't know, do you need to use maybe the comma is not necessary? No, it is. It knows that you're trying to create a new argument, so you can choose from the drop-down menu. That also works if you try, you don't remember how I do a dot plot. Oh yes, it's a spell like this, okay. Please run the next step, and when we are coming back from lunch, we'll go through what is happening here.
What is the meaning of calculating each cluster? What does it calculate?
Oh, you want me to explain it now? You're not hungry? No? Okay. So,
And how is there a cluster zero? What do you mean by cluster zero?
yep, I'll explain in a second. So again, most of the time I give some information what is happening down here. It's true that I may not explain anything, but I tend to do more mistakes when I'm talking than whatever is here. So here we're calling there's a function called find all markers we give in the object. Again, this could be omitted, so I don't have to, this is the same thing. Positional or being explicit, right? This function is gonna go through all the identities and do differential gene expression analysis. So for one identity compared with the rest. Right? So it's saying,
So cluster one will be compared against all other clusters.
yes. In here, I'm telling only report whatever is positively higher in my cluster. I don't care if it's downregulated, don't show that to me. Only show the ones that are positively regulated, upregulated on my cluster, or the cluster that you're currently analyzing. This thing is telling that gene needs to be expressed at least in 25% of the cells. If it's less, ignore the gene. So it means that the gene needs to have a circle of this size in the cluster. If not, don't use it. Oh, I forgot about the threshold. Oh, this is the fold change. Yes, I think this is the fold change. So am I explaining here, right? Yeah, log fold change. So this is the fold change. So if it has a fold change lower than that, don't report it. It's too low. And again, I'm being explicit. You don't have to be explicit because whatever is active is this to say, but I'm being explicit. So what is happening here is taking the identities, which currently the active identity are the clusters, going cluster by cluster and doing differential expression of cluster 0 against the rest, cluster 1 against the rest, and only reporting whatever is upregulated in that cluster. If it's downregulated, don't report it because I don't care. So can you guess what I'm trying to do here? I don't hear you. What? Volcano plots. No. What I'm trying to do here is identify, well, probably I'm telling you here already, cluster-specific markers, right? If you could imagine that if cluster 0 has 10 genes that are regulated, those are the genes that define cluster 0.
The signature gene for that.
Yeah. Yeah, that's a good way to look at it. I think going for downregulated, it doesn't, I don't think, so when you try to identify a cell type, right, you don't say, oh, the cell type has downregulated. You generally say the genes that define a cell type are upregulated. So the goal of this is to help you to identify cell types later on, right? So what cluster, we have a cluster, there were a few clusters with CD3D, right? It should show up on those clusters, but it's a bit dangerous because we are comparing, let's go back. So CD3D is highly expressed in multiple clusters. What final is doing, I'm getting only this cluster and compare it with the rest. So there's a bit danger here that CD3 is also expressed in other clusters too. So let's see what happens. Let's look at it at CD3D if it shows up on the same clusters that we saw up there. Oh, it's on, so we didn't have to wait that much. So you already know what we're doing here, right? Showing the top 6 lines of whatever new object we created. So what we have here, p-value of the differential expression analysis we just performed, the average, right? So we don't have any more a single log-fold2 change because we have a group of cells now, it's a cluster. So this is the average log2 fold change. This is the percentage of cells within the cluster that we are currently testing that express that gene. This is the percentage of cells on the other group that we are comparing, which is the rest, that express that gene. And this is the p-value adjusted, which is the one that we should look at it. And this is the current cluster that we are testing, which is 0. So basically, we are looking at the top 6 genes for cluster 0.
The second cluster would be a monocyte.
Well, this is no second cluster. So this you probably are yeah. So cluster 0, you are estimating that probably is monocytes, probably. So CD14 is a monocyte marker. We find it as a probably uniquely expressed in cluster 0, so maybe cluster 0 are monocytes. Maybe the other is MS4A. It sounds like a also known cluster for something. I don't know. That's all, again, I'm not good at it, but you can Google it and figure out if it's an important one. You can Google that one or that one and see if are defining some cell type. If all of those are monocytes defining markers, you convince me now that cluster 0 are monocytes. Okay, I know. You guys ask questions, I only answer them. Don't blame me.
Hello.
Hi. I think we left it here. I'm not going to make you to do the next exercise. We can do it together. So we can move. I think probably this was the harder exercise, and if I tell you the truth, it's not that useful. Here I was saying to create a dot plot, similar to the one we created up there, right? So, but what we're doing now is taking from the Seurat cluster makers markers SCT, so the top markers of every cluster. We're going to pick the top 2 of every cluster. And out of that, we're going to make a dot plot. So again, I'm not going to make you do this. I think it's harder. If we had time, I'd let you practice and Google stuff, but we don't have much time for Googling now. So basically, I'm asking, let's create a vector of genes that contains all the top 2 genes for every cluster. So if we use the dplyr approach, so this is the name where we store this information, right? This is the object that contains our table. And we're going to use the pipes. So we give the pipe to the next thing. And I'm mentioning that you can use filter, group by, and top n to obtain the final result we want. So the first thing I'm going to do is filter for, I don't think you maybe necessarily need this, but let's keep only the genes that are significant. It's called p-val adjusted. So p-val adjusted. Right, and we want them to be less or equal than 0.05. Okay, now we remove anything that is not significant in that table, if there's anything like that. Then we want to group by, and you can Google what everything does and all the parameters you can do. We're going to group by cluster. So the next operation we're going to do is going to be at cluster level. Cluster is the name of the column, nothing else. Could be any other column. So we're grouping by cluster. So those operations are going to, sorry, I didn't want to do that. The next thing we're going to do is be affecting every cluster. And I said to take the top n is a way to select the top, whatever you want to select. I said 2 genes. And if we look at the arguments for top, there is one called WT. That's, I don't know what it means, but it's called WT. What we're trying to do here is selecting the top 2 of what? How I need to order the data, right? So, and for that, we're going to use the average log2 expression. Thank you. There is one more thing that I didn't specify in this exercise, which is we only want the gene names. So far what we've been doing is filtering, removing anything that is not significant, operate at the cluster levels, and selecting the top 2 of every cluster based on the average expression. But that is going to return us another table. I don't want the table. I just want the gene names. I don't know if we use that function. I think it was mentioned maybe yesterday. There's one called pool, and you give me, it's called gene. Yeah, gene. So give me the gene name. And this is an extra thing I'm going to add just in case, which is just the unique ones. It could happen that the same gene is a top gene in more than one cluster. I just want it once. I don't need to have the name duplicated. I know this would have taken a little bit of time because I want you to search information yourself and all that. So I thought it would be good if we do it together. Also, it's really cool, right? I think it's more readable if we try to do this using dplyr and the pipes, so you see what we do in every step. Don't worry, I'll go down, right? The next thing I want you to do is to use the dot plots. I'm probably asking you, always ask, oh, it's here. Okay, well, sorry. Let me take all this. So I don't have to type, oops. Okay, I'm going to have to check a lot of parentheses here. Let me remove all the, did I said save it into a file or not? Oh no, we didn't need to save it, so no saving, out. Now I need to be sure that parentheses are okay. They are not. So any questions on how I selected the genes? I mean, this wasn't an exercise that useful, but I wanted to show off how you can use filtering on the tables to obtain something in a quick way. Dot plot, we already done a dot plot. Oh yes, it's saved to a PDF. We are not going to save it. We're just going to display it. And I think this should work. Hopefully, no. Why not? Oh, it's called top with uppercase. Should I keep it here? I don't know. It seems it's not important. I mean, you can skip this. It's not a big deal. So now, what we have here, and it's quite obvious, right? So we have the clusters on the left side, and we picked the top genes, the top 2 genes of every cluster. So that's why you see that diagonal lighting up in purple, right? Because we already been selecting those top genes for every cluster. Although some of them, like here, right? Cluster 6 and cluster 13, they probably have the top 2 genes are the same ones, or close. They're going to be top. Not the same ones, but they're pretty close. Maybe this information is useful for classification, for cell typing, I'm not sure. Okay, can we do the next exercise? So one thing you can do to make your life easier, maybe, you could download, did we save an Excel? Yes. You could download the Excel file in your computer, open it, and start classifying the clusters, or assign clusters to a cell type. You want to try? So we're doing manual cell type classification. And there is no wrong or right answer because I haven't done it. So I don't know. Let's give it a try. I'm giving you some different ways you can do that, right? Search for canonical PBMC markers. That could be a way to do it. Open the Excel, check the top markers of every cluster, see if you identify them. Dr. Amit already identified maybe monocytes, I think it was, monocytes. Yep, cluster markers. One thing to confirm your theory is you can visualize them. Once you create a list of markers, you could visualize them. Sorry, what? You have to, there's no Excel reader on the website. So if you do, what is it? I mean, let's do one thing. Why you don't work together in couples or 3 people together? It may be easier, right? So if you group in couples and tie to the side. So once you decide cluster 0 are monocytes, come down here in the next cell and say these are monocytes, which I don't know if they are, but let's assume they are, or whatever you think cluster 0 is. And remember, it could be that cluster 6 and 7, both of them are monocytes. So name them as monocytes, both of them, if you think they are. I don't know what they are. So, or call them, even better, call them monocytes 1 and monocytes 2, just in case.
You have spelled monocytes as monocytes.
Oops, sorry. Well, it's anyway, I don't know if they are. This is an example. I'm going to keep it as a cluster 0 for now. I'm renaming the clusters. I'm renaming the cell types. They're wrong.
We have to do it manually.
Maybe. That's exercise. Or, hold on, let's change the tactics. Why we don't group each two people names a cluster differently, tries to figure out what it's each cluster. Why we do cluster 0 over there, 1, 2 over here, 3, 4, 5, 6, I don't know, that corner 7, 8 around there, 9, 10, 11, 12, okay, 13, and forget about 14, 15, and 16. Let's assume that's something else. Use Google too, right? Google may help you. Or or chat. Or okay, yeah. What I mean, you can try. Give it the top 10s of a cluster, the top 10 genes, and say what I identify as these that have been top 10s in my cluster. Can you tell me what cell type is this? And see what happens. Or give it the whole thing. The whole thing meaning your cluster only. The ones that you think that are the top ones. Maybe 50. Be polite. Say thank you when you finished. And remember, let's say you decided to look at cluster. I'm going to look at the top 10 genes of the cluster. Not all of them are going to be informative of what cell type it is. You're running out of time.
Just testing me out. I'm not doing it.
If you don't find a cluster, the bomb is going to explode. So it's up to you guys.
Okay.
Okay, you already figured out one? Okay, hold on. Hold on your thought. Okay. I don't know. I don't know the answer, so I don't have the right answer. So we're gonna try our best. Okay, hold on, hold on. What's gonna happen at the end? It's gonna explode? Oh yeah, it explodes. Okay, cool. Okay, now that you're here close, so what genes in your cluster 1?
Cluster 1.
What genes you think are important in cluster 1? Just give me CD8A, CD8B, TLR. Look at the screen and let me know that I'm spelling that right. K. Like this? Show me to me, probably it's easier. So we don't have to do the spelling contest. Oh, KLR1. Oh, KLRK1. Okay, I got you. Any other one that you think is important?
Yes, the full CCR7.
CCR7. KLR1K. CRR7. It's like that. CRR or CC?
CCR.
CCCR. Okay, yeah, got you. Okay, parentheses. What the heck? Cluster 1 identify these to be key genes, right? Sorry, we cannot read it, but that is, which one? And CCR7. So you can do plots also to test, right? It looks like, yeah, cluster 1 are highly expressed. I don't know, could it be that there's another cluster? Having all three, there's only cluster 1 that have all three being highly expressed. So maybe there are T cells? T, CD8? Okay. I don't know if let's keep short names, maybe it's easier. So should we name, what did you, cluster 0? What do you think it is? Give me a name. You're convinced they're monocytes? Okay, let's do monocytes. Cluster 1, we said TCD8. I'm going to call it like this for simplification. Cluster 2, what do we have? ChatGPT failed. Okay. ChatGPT. Cluster 3. Another failure. Okay. CT3. Nothing. 5. What is 5? What is cluster 5? Okay, 5. 6. What about 6? Do we have anything? Give me something. Oh, 6 was a harder one. I think it was a tiny one. Yeah, so we're going to go fast. Don't worry about it. We'll figure it out. 7, anything on 7? 8? 9? What do you have? So CD8? Oh, you're giving a gene name? No, I want, what cluster is this? What cell type is this? Sorry. What do you think is the cell type? Okay, CT8. Cluster 9? NK cells. 8 NK cells. Okay. NK9? NK9? Oh, okay. I thought you want me to name them NK9 cells or something. Okay. You're saying these one are the NK cells. Okay. Let's keep it short. 10 B cells. 11. 11 status 11. 12. Okay, it could be. So what? Monocytes, macrophages.
Struct 10 is B lymphocytes.
8 B lymphocytes.
No, no. 10 is plus 10 is B lymphocytes.
Okay, B lymph. I know you're not going to be such in a rush. Let's keep it like this and we move on. So we decided that's going to be our clusters. I don't know if we need to merge or not merge. That's something we haven't considered. It looks like we may have multiple B cells, so it could have good idea maybe merging them, maybe. For now, we're going to keep the same structure. So it's pretty explicit what this thing does, right? We are renaming our Seurat object with new clusters IDs. So we're going to take this dictionary that we have here that has the current clusters, and we're going to rename them with our own names. I'm also doing one extra thing, which is in here, I'm counting how many of the cells we have for each cluster. It's a bit flat, but so it looks like we have the macrophages, monocytes, right?
Cluster 2 is also which monocyte? Cluster 2 is monocyte.
Two. Oh, ChatGPT is also monocytes. It could be next to each other, right? That could be a good merging. Do you want me to merge them so we see how it happens? We're going to keep the same name. So was cluster 2 also macrophages? That's what we're going to say. So exactly the same thing. They're what, sorry? So T cells. Which one? 13 are also T CD8s. That's how we name them, right? Somewhere. Okay. That's it. We don't have to rename anything else. That's good enough, I think. I like that you get excited about this exercise. That's good. So now we merge a cluster, right? Yeah, we don't have those two clusters that we have here, that it's only one, and I don't remember. We probably merged something down here, right? Those clusters that we merged, the CD8s, they got merged. So this is quite common. Again, our clustering doesn't need to represent cell types directly. It's not one-to-one. It depends on a lot of different things. If you want to subclassify the TCD8s, you could use those two separations that we created. But if not, we could merge them. Questions? Well, here in the next paragraph, I'm just talking about the challenge. You already noticed that we can have really tiny clusters that probably we need to merge with bigger ones.
But there must be inbuilt tools we can do that for us.
That's going to be next, yes. But even the automatic tools, they have problems too. So instead of us making that decision, there are tools available, like single R is one of them. There are a bunch of them. Actually, I think Seurat is pushing for one called Azimuth. So most of these tools depends on some reference database. They need something to be able to make that association, right? And you can do it at two different levels. You could take a cluster and try to classify a cluster as something, or you could classify a single cell as something, and then your clustering, you can throw it away because it's not going to match. If you start classifying cells individually, whatever the algorithm tells you it is, that's going to be the new clustering. The other way is assume that your clustering is good enough and then try to classify your clusters, cluster by cluster. So in single R, so we use CellDex to access some database. You have a bunch of them available. I'm not an expert in databases, so I don't know exactly which one is going to be better. I think I choose this one instead of going for a PBMs specific. I thought this one would give you better. It would be simpler to show than if we go for one that is really specific. But I mean, if you want, you can try. If you comment these and uncomment one of these other ones, you can try different data sets with the next process and see how the difference is. If you notice, remember that I told you this. So in single R, we are not using the SC transform data. And luckily, we've done the normalization that we did initially. We're going to use that assay for this.
I have one query.
And another thing we're doing this. Sorry, yes, I need to explain more details. Single R doesn't like the Seurat object. So the format that Seurat object has, it doesn't like it. So we are transforming the Seurat object to one thing that is called single cell experiment format. And we are taking only the RNA assay. So we are forgetting the SCT. So this is like a Seurat object with only this. It's similar. Let's assume that. So now we're going to run single R with this object that we created, which is, imagine is like a Seurat object, and one of these references. I picked that one here, which is more generic. So for the classification, we're going to use the clusters. We're telling single R, use our clusters to classify. It's going to take the whole cluster and try to figure out what is that thing, right? The whole cluster. I think single R can operate at the single cell level, but we are not going to do it. There's, again, Azimuth, you have different Azimuth versions. They operate at single cell level too. Some references, they have different levels of annotation. So they go from level 1, and they tell you, the higher level hierarchies, right? Level 1 is going to be, they're going to take your T cells, all the T cells. It doesn't matter if they're subtypes or not. They have a classify more details. And level 3, they classify even more details. So I'm assuming, I'm not sure what it contains of these, or this specific one and what the levels are, but some of the databases that you may use, they can have different sub-levels. So I've never used these before this workshop. So I don't know many details. I use other tools like Azimuth. I use Azimuth sometimes. So, but I thought this would be, I guess, easier to understand what we're doing. We're accessing one database, but we can access these other databases that are available. I'm not going to go into details because I don't know them, if I tell you the truth. But one thing, so these are, they're not going to give you the truth, right? They're going to do some statistical analysis. They're going to give you some sort of a score and tell you the most probable cell type is this. They probably have other type of, so let's say, some of them they're going to tell you, I'm 60% sure this is a monocyte. But what about the 40% left, right? I'm also thinking maybe it's a B cell or whatever. So sometimes you may get a single label out of a program like this, But you need to think that there's a confidence of interval inside of that estimation. It's not the truth. They're not giving you a single answer a lot of times. It could be more than one answer. I'm explaining that idea a little bit, yeah?
For T cell, it is not specifying that it is CD8 or CD4 or...
No, that depends on two different things. How good is your reference? So if you're working with a reference of primary cells in your body, they're not going to be really specific. If you go to a reference that is specific for PBMCs, maybe you get more details. So your choice of reference is going to define how good is going to be your final goal too. But again, these softwares, they're not, first, there are two things. They may be incorrect, and they're always incorrect at some point. They're not always 100% right. And second, they always have they give you some confident value about how confident they are that every cluster is whatever they think it is. They are. So this is our results. It's been quite generic, right? We give them monocytes. I think we were right on that one, right? We said monocytes, macrophages, so this was right. Did we set B cells on top? Yes? Okay, that's good. Definitely we said these were T cells. I don't know what we said about these ones, but it classifies everything as T cells. Did we set it with NK? So we fail in that. We may fail on that. We're assuming that Singular is doing a good job, but we don't know. Yep? So if you change the database of reference, you may get different results. So if you try the specific one, I don't know, let's see what happens. Oh, there's the azimuth. I didn't know I added. Okay, this was the reference for PBMCs. Is it a bit better in the classification? I guess we want more detail. This is giving us a little bit more detail. NK cells, so even if we change the reference, they insist these are NK cells. CD8 positives, that's what we said, right? This group was. So we were right about that one. Still monocytes, dendritic cells. I don't know if it makes sense. It makes sense?
Yes.
Yeah, please.
This is by R and okay, but what you have done previously want to identify the cluster, you didn't get actually. So can you explain it again?
Which part?
From Excel.
How to identify the cluster?
From Excel, right?
From here?
No, actually some people didn't get that. You have to download that Excel and look at the genes and try to guess.
Yes, that was the goal. Download the Excel and from that table try to guess what it was. Yes, that was the idea. From the cluster-specific markers, I would call them, try to figure out what the... Yeah.
Actually on the Mac it is not opening.
It's not opening?
No, I have downloaded it but it's not opening. Numbers पे खुल नहीं रहा है.
अच्छा वर्ड डॉक्यूमेंट होगा मैंने माइक्रोसॉफ्ट वर्ड है ना
मेरे Mac पे अपलोड हो जा रहा है पर वो ओपन नहीं.
हाँ वो माइक्रोसॉफ्ट वर्ड नहीं होगा माइक्रोसॉफ्ट
नहीं मेरे पे नंबर्स है ना तो उसको एक्सेल को नंबर्स में मेरा कुछ मिसमैच करके बोल ही नहीं रहा है.
So we didn't get, NK cells. We placed them here, the NK cells. We were wrong. In theory, we are wrong about the NK cells. So another tool, that's the one I was mentioning, Azimuth. If we run this, if you want, you don't have to run it. This is just the last exercise. We give indirectly the Seurat object, and let's see what we get out of it. So this is one of those tools that there are different levels of classification. So from generic, middle, I think there are 3 levels to detail. I think this is going to be way too much. I don't know how it's going to look like, but I'm assuming
It looks quite crowded.
it looks really crowded. Yeah.
But it has classified even the B cells into different subsets.
Okay.
Naive, memory.
So in this case, is ignoring our clustering. It's throwing away the clustering and goes cell by cell and trying to classify the cell into something. Oh, it's accurate? You are the biologist? I don't know if you're a biologist. Maybe you are the biologist, you need to tell me that. So it is using, let's say, a very big data.
Sometimes the labels are hiding the cluster itself.
I know. Then we need to play with the labels and see. I mean, we can change these and go a level up maybe, so we get less classification. Or then you can manipulate the table and say, show me only this group, so I can see where they are instead of showing me everything. Did it match our previous expectations? Like NK cells are still being NK cells? B match. Okay. Oh, it finished, right? So CD8, it matched. So this one also CD4. B's. Yeah, NK cells are here, right? They're still here. Yeah. So here gives a mixed. Definitely monocytes and something else. Oh, a different type of monocyte. So yeah. Yep. Questions? Generally, we do both. We do it the other way around. So first we do something like this, and then let's say the investigator confirms whatever they think is correct. We generally don't need everything on the analysis, right? People wants to focus on a few cell types and then ignore the rest. So
some cells are very few in number, like plasma cell in the periphery will be very few.
yeah, I don't know what it is here. I don't know. Is expected to have only a few plasma cells on PBMCs?
Expected very, very few.
Okay, so it's.
PDCs also will be very few.
I don't know what's a PDC. I have no clue.
It's a plasma cytoid dendritic cell.
Okay.
And
So
also you got a megakaryocyte also. Megakaryocyte is in the marrow.
it's here, somewhere behind this label, right?
So it is a megakaryocyte.
Okay.
So normally you should not see a megakaryocyte in the peripheral blood.
So let's do. So it's GG repel. Sorry, how is it spelled? G repel. It's not called like this. How we use it? Okay, it doesn't matter. And as always, remember at the end of your notebook, you can record your session, save it in a file. So any questions?
No, but if you want to see that megakaryocyte, it's a separate cluster, is it? Like which cluster number is it?
Well, azimuth, again, ignores your clustering.
It doesn't tell you where the cluster was.
No, it's so So when you run azimuth, you are classifying cells. At that point, you're clustering. Whatever you've done previously is gone.
So the cluster information is not there now.
No, it's not there, and it's never been there for azimuth. So when you provide CIRAD to azimuth, your classification of clusters is not taken into account for anything. It goes cell by cell. So in the previous exercise, what we did is single R takes a whole cluster and tries to tell you what it is. In this case, it takes only one cell, cell by cell.
I have one question. This is one sample data, right?
Yes.
How the log to fold chains are coming? What we are comparing? What with what? The genes they are giving the log to fold chain.
Yes. So the log to fold change we got earlier, it was comparing every cluster with the rest.
Okay.
So cluster 0 with all the other cluster.
The rest of
Yes. So that's how we identify what I call cluster-specific genes, right? They're not 100% specific because sometimes you see them in different clusters, but that's the idea.
But how is azimuth so fast in the sense that Surrey is going to do it cluster by cluster, but
Yeah.
this is going to do it by cell?
I know. I don't know.
So
I don't know the details.
return the outputs
Yep.
quite fast.
Yeah. This is it connects to the internet. By the way, you cannot do this without internet connection.
So if I'm going to manually evaluate, right, I just wanted to confirm how I manually evaluated the
Yeah.
cluster. Basically I should sort them by the log, see whatever, and take whatever is the highest popular genes, and then compare them to whatever canonical
Yep. Yeah. And if you probably Google PBMC canonical markers, you're probably going to get a table.
I got a whole list.
Yeah.
So that's how.
But sometimes there are clusters that are really hard to classify because the clustering is going to maybe create a small cluster and there's not enough genes to classify. There's always complications. It's not that easy. So for a sample like this, there's not much less left to do, right? We classify the genes. I don't know. We could do a comparison between T cells if we want to.
Will we do the micro exercise also?
We could. It's depending on what we want to do next.
Some parts we can do the micro exercise.
We can try, yeah. So before I move on, any questions about this notebook? One thing I can do is try to go fast through the next one in case. What do you think, Bonnie?
We'll do the functional and then you come back after the talk and then they want.
Yes? Okay. So we're going to take a rest now from single cell and he's going to do functional analysis. Oh, no, sorry. As always, Bonnie is not prepared on time.
He was doing it in the morning.
Yeah, exactly. He had months to prepare for this.
This workshop is actually pretty light, okay, in its content. So if you ever get a chance to attend some of the Cold Spring Harbor's workshops, usually they are 3 weeks long workshop, okay, and
So I did one of them for functional genomics statistics. Yeah, so they have this one, the statistics for functional genomics. So it's their bioinformatics basically, 3 weeks. So 3 weeks, every day it will start at 9 o'clock, then go all the way till 5 o'clock, then you will get 2 hours break, then 7 to night 12. And this you are doing like 3 weeks stretch, like not Saturday, Sunday also there is no break. Only one Sunday they will take you out for like a team building kind of a stuff, which is too fun. I actually took this workshop and then I came and slept for like 3 days straight. Like and the worst part is this 7 o'clock on, so they will 5 o'clock they will have a like break, 6 o'clock they will serve dinner, and then 7 o'clock onwards it's hands on, like you will whatever they taught you will be doing those exercises, and the course people will be there helping you to like, so basically whatever you learn in the morning you will have exercises to do, and this, and there will be alcohol actually. So people start drinking from like 7 and everybody is like
Till morning 7.
no, so till 12. So 12 is like normal, and then after that there is a bar in Cold Spring Harbor. So where Watson used to come and we used to meet Watson there. That people will actually stay there till like 2 o'clock, 3 o'clock, depends upon how long they want to be. There was a faculty, he was a crystallographer in University of Florida, and he at that time I was like in 35 or so. He was in 50s, and he was doing straight 5 o'clock every day in day out, like for 21 days straight, and just 4 hours sleep. During that time he also did his analysis also. He brought a data with him. Basically he learned everything and analysis, and he analyzed his own data, everything ready for a paper for him. Just, and same thing, like they have 5-day conferences. Again, that every single day it will start at 9 and end at 12:30 in the night. Main keynote speakers are actually scheduled for like speak at 10:30 in the night. The last talk will be the keynote, and they will be like some very big shot and everybody wants to listen. That means you cannot go anywhere. And again, after 5 days of conference, you think like, okay, I know I don't want to live anymore. Just let me die. Allow me to die. So, yeah. Cold Spring Harbor will give you travel grants. Apply for that. Those workshops are amazing. I mean, if you get into one of those workshops, I will show you their web page actually. They have a whole series of there is one on genome also is there, but no, their workshops are actually year round fixtures. So and it's actually permanent ones. Like
I did one which was online during COVID.
so, yeah, you should attend one of those workshops. It depends upon how you want to use it. I mean, end of the day, you know, it's fun. I mean, once you come back from there, you will be completely changed person. Like, this is Cold Spring Harbor. I will show you the website. You know. So the good thing about Cold Spring Harbor, so it's a mecca of molecular biology. All initial molecular biology discoveries were done in Cold Spring Harbor. Like jumping gene, Barbara McClintock was there. Hershey and Chase experiment, that was done in Cold Spring. So every single then microRNA discovery by Gregory Hannan, all those. So every single big molecular biology discoveries were done from Cold Spring Harbor. And when you go there, if you are a molecular biologist, you will just feel goosebumps. Like, oh my God, I studied these guys, and there's a house where. Yes, in Long Island. There's a house which is nobody's using. It used to be Barbara McClintock's house. So it's a quarters. So she first proposed that genes can jump. So transposon, she said, and nobody actually believed her. And she almost like secluded herself for a long time before it was rediscovered. And so that house is still there, and it's beautiful. It's a coastline, and you can have fun. I mean, that place is fun. I actually took my son and wife recently when we were to Cold Spring Harbor, Long Island, and so I was actually told, okay, let's go there and show this place to them. It's a beautiful place also to just visit also. but it's too nerdy to take wife and son to a research lab, I know that.
So if you want to induct your children into research.
No, he is actually, no. So my one of the biggest challenge for most of the gene expression analysis is what does that means. Okay, you have 505 and now you have another situation where based on whatever cutoffs you put like 2-fold change, 3-fold change or p-value cutoff, whatever cutoff you put, you will get different numbers of genes and different set of genes. Okay, the question is what that data means actually. So suppose if I have a differential gene expression table and I am just doing a p-value cutoff, I will get 1500 genes. But same time you want also to add a fold change value of 2-fold or more. That means now my numbers will be 600. Somebody else wants some other cutoff, so they will get a different values. But the question is, none of this value really tells us anything about the biology of the system that you are working on. So why it is important? So to do that, you need to understand an idea called as Waddington's landscape. This idea was actually proposed long back, even before genes were discovered, okay, in 1936. And this is called as Waddington's landscape for it's Waddington's epigenetic landscape basically. What is really happening here in this, that you have a cell, okay, think about a cell. When cell is actually rolling through a hill, then each point, like this is a point where cell has a choice to make, like whether it wants to go this side or this side. And one of the example, you are immunologist, one of the classic example is PU1 expression. Then when the cell lineage determination happens, the PU1 expression or non-expression will define which lineage it will go. So similarly, this cell will decide at this point that whether it needs to take this path or this path. And this is every such force, cell will be deciding that, making that decision, and it will keep rolling to the bottom of this landscape. And when that bottom of the landscape will be differentiated, terminally differentiated cells. So since long, like almost till 2005, the idea was that cells once reach its terminal differentiated state, it cannot go back to its original state of or pluripotent state, or where it came back from. But then a classic experiment by Yamanaka proved that no, you can actually revert this Waddington landscape and you can go back into the you can actually go back in this landscape. So that was the biggest discovery. Then what happened? People started actually differentiating. They started converting differentiated cells into its original pluripotent state, and then they do a transdifferentiation. Transdifferentiation essentially means that you started with a neuronal cell, you brought it back into stem cells, and then you actually differentiate into muscle cells. So this is an example of transdifferentiation. You can also transdifferentiate cell from one cell fate to other fate, which is not that easy or trivial, but it can be done. So basic idea here is, what I am trying to actually enforce, that each cell actually possesses a particular cell state, and that cell state is defined by the gene regulatory network underlying this. So if you think about it as a tent, what is holding each point of the tent, those wires that actually you pull for the tent, those tents are actually the gene expressions, the set of genes that are being expressed. So any gene in isolation, there is nothing called as lone wolf or genius or prodigy in case of genes. There is nothing like that. Genes always works in group. They works in networks. And if a gene is changing, if it is not changing anything else downstream, then that expression difference has no meaning. Which means to know that what is that combined effect of some perturbation, you need to know what kind of functional grouping these genes are falling into. So all the genes in a differential gene expression table will not fall into one or other functional tail, but there will be a group of genes, those will be falling in a group, functional group. And the whole idea of this class is to understand that how you really do that functional analysis and find functional grouping and what are the different ways you can do that. So let's first do a little bit of theory on this. So one of the common way to analyze gene expression data is through gene ontology. Okay, so gene ontology, or which you are very familiarly known as Go terms, they are structured vocabularies. What is mean that genes are arranged in various levels, and we'll see some of them, and then they are structured through higher level to lower level. Like one of the best example is that genes, all the genes that are present in a biological processes or do some biological stuff. So that is a biological processes. Now it's a broad term, so that is a higher in the hierarchy. And then you have further you go in various levels, it gets more finer and finer. And at the lowest level, you are actually going to find a term which is very specific and will have a very narrow definition. So this is what actually the Go ontology means. So in general, Go ontology consists of three various broad categories. One is called as biological processes, second is molecular function, and third is cellular component. So biological process, as the name implies, it usually deals with pathways and processes that actually defines biology. Okay. The molecular function is basically activity at a molecular level, and one of the example is transcription factors. The best example is that. So if there are certain group of genes that act as a transcription factor, there are certain genes that are kinases. So these are the molecular function, because the kinases function is to phosphorylate. So that means you will be there are ubiquitins are there. So different classes of ubiquitins are there. So that is one kind of a molecular function. Then you have cellular component. So cellular component is the proteins that form the part of the cellular, subcellular components. Like cells, those are associated, proteins that are associated or genes that are associated with Golgi body, or something that is associated with mitochondria or chlorophyll. All this, those are actually comes under the cellular component. So basically same set of genes, you are classifying them under various things, and you are and all the genes need not have all the three functions. Okay, some genes may have only involved in biological processes, some will have only cellular function, but some may have multiple roles also. So this is actually a nice way to interpret the data. So basically it works as a way of parent and child organization. So there is a parent level is there, below that the child will be there, then there is a grandchild will be there. So it goes into that kind of a structure. Okay. So it can be either like it is a relationship between this is A is a part of, or it regulates. So basically it's either it is regulating, or it is part of that process, or it is the process like that. And you can actually do the hierarchical progression of gene function studies using Go term. Okay. Now if you look at hierarchy, I know that this is fonts are small, but here you see there is a biological process, and then it's involved in regulation, then regulation of biological process or cell process. And so basically what you're doing, you are actually structuring a bigger biological process into its parent-child relationship, and finally you are coming to this term which is very specific, which is negative regulation of, say, programmed cell death. So at that stage, when you are actually crossed all those hierarchies and came at the lowest level of the data set, that means now that term is very specific. This is only related to negative regulation. So even not regulation of programmed cell death, it is only negative regulation of programmed cell. So you can go up to that finer level. Most of the analysis what we are going to see today we are allowing program to choose its own level. So program will actually go through all the levels and find out a level which is most suitable for it. But then you can also enforce levels. Levels are between 1 to 7, and you can say level is equal to 3, then it will actually take the third level of that hierarchy and do the enrichment analysis for you for that. So you can also enforce levels, but in generally the programs automatically allows you to choose between the levels. Okay. Now the goal of a statistical goal of a GO term analysis is to identify GO terms or gene ontology terms that are significantly enriched in your data set. And then how you do it, you do Fisher's test or chi-square test, and depends upon what kind of data sets you are using. Most often it is the Fisher test or hypogeometric test is being used, and then you do a multiple testing correction or which is basically either Benjamin-Hochberg or Bonferroni correction. I actually tried to explain this multiple to some people yesterday. The idea here is that if you are asking same question 1000 times, there is a chance that you are going to get a false positive answer. So in this kind of analysis, every pathway you are taking or every ontology term you are taking and you are asking, are you differentially enriched between my sample and the universe or not? If that is the question you are asking. When you ask this question repeatedly, there is a chance that you will get a false positive response, saying like somebody will randomly say, yes, I am. So to do that, you need to correct. One of the best example which I was talking to some of the people yesterday, the way I gave that they told me that Manpreet is the name which is most common name in Chandigarh. So if that is the case, Manpreet is the most popular name in Chandigarh, and I want to meet Manpreet, and I don't know who is Manpreet, and I only know that okay, there's a person called as Manpreet, and she is from PGI. Now my choice is like if I came to Chandigarh and every single person I meet, so I just know she's a girl, or maybe Manpreet can be a boy also, no? Yeah, it's a gender neutral name, so that makes it more complicated. So every person in Chandigarh, I will go one after another and ask, are you Manpreet? Are you Manpreet? Randomly there will be a person who will say, yes, I am Manpreet, because that person is Manpreet. This is the most popular name in Chandigarh, so it may be boy or girl. At that point, if it is a boy who is actually responding or man is responding, then I know that okay, it's possible that it's a false negative actually. But at that, and then if suppose happen to be a girl, then I need to actually also add a second criteria, which is, then I will ask, are you from PGI? Then she say, okay, if not, I am not from PGI. If actually that person is also happen to be for PGI, then I need to have another criteria to figure out the Manpreet I want to meet. So this way, when we are actually doing same test, when we are doing thousands of times we are asking same question, then there is a chance and random chance that you will get a false positive, and you need to correct it for that false positive. And there are two different ways to do that statistically. One is called as Benjamin-Hochberg correction, and second is called as Bonferroni correction. Unfortunately, both of these corrections are harsh on the data sets. Okay, so that is why what happened. There are chances like you may remove some of the true positive also in this correction process, and but this is actually a very conservative way of doing it. What people found that there is an alternate way to do such a corrections, and which is called as FDR or false discovery rate. In case of false discovery rate, what you are doing, you are first measuring the distribution of p-value, and based on that you are creating a statistics for false discovery rate, and that will give you a little less conservative way of estimating the correction, and then that's how you can do a multiple hypothesis testing. So in case of GO term analysis, both ways, FDR as well as Bonferroni and Benjamin-Hochberg, both ways actually people are doing. Some places you may say a value called as Q value. Q value is actually related to FDR. So some people say that as FDR, some people say Q value, but if you are asking this to a statistician, they will say I am wrong. So but in fact, you can actually use for a practical purposes, you can use those two terms interchangeably. Okay. Now, how to do enrichment analysis for GO term? So idea is very simple. There is something called as universe. Okay. So GO term is a curated database. So somebody is really reading a paper manually and curate that database. And when I was in Jackson lab, the person who used to sit next to me was doing this. His job was such a, like whole day he will sit in a room and his only job is to read paper, and based on that he will curate that database. So you think about how boring that job is, but he was happy about a lot. He was doing that. So anyway, so you have a universe. So all the genes are not there in Go database yet, and that is one of the major limitations of such databases. So you have only 6,000 some genes are there. When last time I checked, 6,800 genes something. So that is the universe. Okay. Now you have a differential expression table that you, whatever criteria you actually had, so based on that you created a differential expression table. Now that gene has some number of genes. Now what you are asking, that you take one pathway, say suppose you are taking wind signaling as a pathway. You will ask how many genes are there related to wind signaling in my universe. So which is this X, okay. That means you will actually subtract X to M. M is actually the total number of genes that is there in the universe. So you subtract M minus X and you will get this Y value, which is the number of genes in my universe which is not wind. Same thing you do with your differential data set. You will ask how many genes are there in my differential gene expression table which is related to wind, and so then you minus that with differential expressed gene, then you will get Q value which is not related to wind. And now you actually do a F test, because this is a 2 by 2 contingency table, and you just do a F test and you will get a P value, and that will tell you that whether this is significant or not. And when you are calculating fold change, you are basically calculating X divided by Y and then P divided by Y, and that ratio if you take, that is the fold change value for your Go term. So this is the process that being done, and this is done for every single term that is there in the Go database or KEGG pathway database. Any database, whatever number of terms are there, the program will do that in the backend for each term, and they will calculate the P value, they will calculate and then they will do a correction for the P value, because now you are doing this test hundreds of times, different pathways that need a correction, and once they are corrected, they will report the based on P adjusted value which is significant. Okay. Now, so in generally what happened, the input list for most of the analysis which uses over-representation analysis, the input genes are ORA, or input lists are significance genes. So you will make a criteria for significance and you will put them, and then you will compare these frequencies, and then you report based on adjusted P value. So there are many such tools are there. Manpreet is going to talk about David, but meanwhile I will be talking about cluster profiler, which based on R, and then there are Python based other tools are also there. In case of gene enrichment analysis, what happens, one of the key question is why we should have a criteria at all, like two-fold change. What is the sanctity of such a fold change biologically? I mean, do really cells care, like if a gene is expressed 2 times more or 3 times more or 5 times more? No, they don't, really they don't. But this two-fold change idea came from cancer biology. So all the microarray research started with cancer biology. Okay, so when cancer biology data were being produced, they have large number of genes which are getting changed in such experiments. So if they set up a two-fold change criteria, still they have enough number of genes to further analyze downstream and then get some kind of a biological insight into that. So that is how this two-fold arbitrary cutoff came. But that stayed as if like everybody you ask, they have no idea why they are using two-fold, but everybody is using two-fold cutoff. So there is nothing like such a cutoff. Biological system works with a small incremental effect, and if you ever actually worked with deletions like loss of function mutants in mouse and others, you will see the changes, that gene expression changes are very small, not very big. Very few genes actually changes huge, but most of the time the functional genes changes make small changes. So in case of this kind of gene set enrichment analysis, rather than doing a cutoff based on log two-fold change or something like that, just you take all the significant genes based on p-value, and then you go and put all those genes together in the analysis. Idea is that even the smallest change in a gene expression, but it is a part of a larger set of a network, that means that change is significant. So now you are not relying your observation based on the magnitude of change, but even the smallest change can be considered as a biologically significant. So
What should we take? P-value or adjusted value?
adjusted, always adjusted p-value. P-value, especially with any kind of this kind of data, p-value, unless it is not corrected for my multiple hypothesis testing, those p-value has no meaning. Okay. So basically, this kind of analysis will avoid any kind of arbitrary threshold, and it also actually includes some kind of a permutation testing, and we will try to see GSE if we could run them from at this time. So one of the things that you always think about when you are doing this kind of analysis, you should always stick to one kind of data sets. You can do analysis using either various kind of IDs like ensemble ID, gene symbol, and interest IDs, everything. You can just use any kind of ID, but whenever you are doing analysis, stick to one kind of ID, and then every time use that ID for analysis. You can also create a background set, which is a good practice. You can always do that. Look at actually always use the updated GoTerm analysis data set. One of the challenges is that if you are doing a GoTerm analysis like now, and if you do after a one year or so, the new terms might be added, new genes might be there. So which means that every time you do this analysis, not like in a day or two, but then if it is done 6 months or 8 months after, you tend to see a difference in your results. So you should always be aware of that, and so that it's always a good practice to redo even the old analysis and make sure like you are, when you are comparing your data, then it's all done together. Okay. The other thing is like most of this analysis, they will give you some random pathways also. And the reason why that happens because same genes might be involved in some other pathways, and I will actually come back to that sometime later, but if I find time I will tell you why. So you have to also think about the biological question that you are interested in. If you are working on a heart development thing and you are finding a viral pathway in that, you know that that is not real biological. So you can definitely omit that based on your domain knowledge. And so basically at some point when you have that pathway in your hand, it's always important, rather than just reporting what is computer is giving, rather than think about it, see like if it makes sense to you, or what are those pathways in your significance list gel to your question that you are asking, and can you test those hypotheses. Okay. So one of the major problems a lot of time happens is the outdated GoTerm. We already spoke about it, and then ignoring multiple corrections. So you have to always use adjusted p-value, not the p-value. Then you also look at that how to avoid this issue, over interpreting generic or redundant terms as we go. And then also it's important to validate your finding from an independent way. Like suppose you get a cardiomyopathy related pathway as enriched one in a mouse experiment, you need to check for that function, like is mice is actually getting those phenotypes or not. So okay, is the pathways that you are getting, okay. Then finally we are actually this is basically what I was talking about. It's a structured data sets, statistical enrichment is done, and then you need to combine pathways and other things with. One of the other pathways which very popular among many of this is KEK pathway, which is basically for signaling pathway. The good thing about KEK pathway is that it's a hierarchical in nature, and it shows you what is the impact of each of this gene over a downstream gene. So this is a KEK pathway, and one of the major bottleneck for many of this analysis is that there is no such hierarchical relationship exists in this analysis, because you remember like how we are doing this analysis, we are actually asking if this is present or absent. Here actually we don't have a weightage, so I can actually quote you two people. One actually Allama Iqbal, he has a very famous Urdu share कि जमहूरियत वो हुकूमत है जहाँ बंदों को गिना करते हैं तौला नहीं करते. So basically what he was saying, that in a democracy you weigh you count people, you don't weigh. And exactly that is what we are doing in Go term. We are not actually weighing the impact of those genes, rather than we are just counting them. So the impact of this gene, which is far upstream like TNF gene, is actually way more pronounced than a gene in the periphery of this network. So if you have a perturbation in this gene like TNF or trail, you will have a bigger impact over the downstream cascade network. So, but none of this analysis, enrichment analysis or aura, actually take into that account. And if possible, I would like to show you this one. This is called as PIA, or signaling pathway impact analysis, where what you are able to do, if two genes, say this is a network of gene, then if you have these two genes have changed its expression in your differential gene expression, which means they are just lone wolf. There are random genes that are actually changing. But suppose if A is changed and B also changed, then that means you know that the impact of change of A is actually passing through B. So you can actually predict the perturbation effect of this. So any of these two genes perturbed, you know that what is going to happen to the further downstream pathway, and you can make a precise calculation for the perturbation and create a p-value. And when you actually create a pathway and enrichment analysis based on such, when you are including the perturbation and weight of the gene in your analysis, and which SPIA does, you will be able to incorporate that also. And then finally, what you also can do, you can predict that what is going to happen with the pathway. So one thing is about the gene, that whether gene is going up or down, but you can also say end result of this perturbation will be that, like say Wnt signaling, if I get Wnt signaling is inhibited. So that inhibition or activation part you will get. In that case, once you have that kind of a interpretation, then you can go back and design your next experiment, where if suppose Wnt pathways are inhibited, then use a Wnt activator and show that you can rescue the phenotype. So that means you have a functional validation now based on that kind of. Okay. So I think this is the presentation I want to give, and let's start with. We did not send that. Francis, can they access those notebooks there also? No. Okay. Okay. You open the fourth day.
So when we select genes by the p-value, right? We select p-value 0.0. That's quite common. What it means? See, we select some intermittent genes, but the 0.05, that 5%.
The chances of selecting it
is the chance of type 1 error. What is the type 1 error? Means the probability of having a false discovery. So you're saying if you do that and you have 20,000 genes, 5% of 20,000 people do that in 1,000 genes are false. You're going to have a significant p-value, right? They're going to be false discoveries. So I think it's good enough reason to assume that you're going to have to correct. If not, you have 1,000 genes that you're going to be selecting that are going to be false discoveries.
Okay. So.
Sorry, did you see the new file? I put a new file in the folder. Let me know if you don't see it.
The name of the file?
Functional something. Functional analysis is showing up.
Yeah.
Not opening.
They have the combined D data set also there?
No, I know. Okay, let me give you a second.
By the time Francis is putting there, I will just show you Cold Spring Harbor. So this is CSHL. So this is called as Cold Spring Harbor Laboratories. It's in Long Island. And if you go here, I believe yeah. If you go to events, there is something called as meeting and courses. And it's a lot of meetings and courses are there. If you go to courses, I'm just showing you all courses. So see. See here you have tutorials in genomics and bioinformatics, RNA-seq analysis, which is from November 9 to November 11. So I think you are a little late for this course. Advanced sequencing technology and bioinformatics analysis, again that is same time. Scientific writing retreat, computational genomics, which is in December, methods in cancer neuroscience, cryo-electron microscopy, quantitative imaging, cell and developmental biology of Xenopus. Like that, you can see like large number of courses, very specific, like ion channels in synaptic and neuronal circuits, a very specific course. Mouse development, which is a very general course. Then this is the one which we go, I think I went for, statistical analysis for genome scale data. They used to call us statistical analysis of functional genomics. The pancreatic cancer, then brain tumors, advanced technology in molecular neurosciences, Drosophila neurobiology. So very interesting advanced topics. They have 2 to 3 weeks. Some of them are 1 week courses also, but most of them are 3 weeks. And you can find that here. And then if you go to meetings, and this is conducted every year, not like 1 year. Then you have like all this kind of meetings are there for them. Like single cell analysis meeting is there. Then you have genome informatics, zebrafish neurobiology, plant genome system biology engineering. So these courses and meetings are very good. And they also give you, students especially, they give travel grants. And DBT also have something called as Women in Science, and they actually give travel grants through Women in Science, so you can actually get that through DBT also. Yeah, you can do same thing here. Basically, once you have the, it's only using the gene IDs, okay. So
for this SPIA, what is there any other software where we can get those in?
no, this is SPR needs to be done on bioconductor only, R only. I think the Ingenuity software from Quadrant, I think that uses SPR as a backend. Yeah, they are paid, but that is what actually they ask me.
What is the difference between what we do, that the string pathways and cytoscape?
I don't know. I don't really believe any of those things, strings and cytoscape. I don't know why. What the?
Way of, making your data look jazzy.
Yeah, I mean, none of those makes any sense. Ever they made any sense to me, unfortunately, to be honest with you.
So they also do show some nodes and the highest specificity.
And I don't know. I mean, I have my own problems. Probably wrong. Probably I'm a priest in a wrong church. No. It's not about you. It's about actually, you are right, like a lot of people are using it. So it doesn't matter whether I believe or not. Okay. It's simply like even. Yeah, but it never made sense to me because I never understood that what part of the biology they are trying to. Yes. No, but my problem was mostly like, I could not figure out that how biologically that is even possible. Like you have differentially expressed gene, and then now you are saying like they are part of a network. So those two things are very different things. Okay, network of, because I'm a biochemist also by training, so the network of a gene networks, like when it is interacting in a network, it usually, there's only two kinds of relationship. That one region regulating other, and other region regulating other. Those things you can capture through most of this kind of analysis via Gotram or anything like that. But then what they are talking about is like if it is a part of a larger like cytoscape and all this, it's complicated. I don't know. I always thought that there is some biology missing there, and I could not spin my head.
One is involved in so many times that it's the same.
It is actually what say. So that you have to think. So that you have to think from evolution. Okay. So
It's ready, Bob.
it's ready. So gene regulatory networks, basic. Okay, guys, I have a question for you. Okay. A bit of a philosophical question, but I want to know that what do you guys think about it. So one of the argument for Darwinian evolution is that the genes are conserved. So some or other form, when you look from single cellular organism to multicellular organism, the same genes are actually conserved. So tell me one thing, why do you think like genes should be conserved? What is the compulsion of genes to be conserved? I mean, there are 20,000 genes. They always can, lose or gain or make a new gene and can do same thing. But genes are conserved. And why they are conserved? Why you think there is a inherent compulsion in evolution for genes to be conserved?
Otherwise, there will be so much change over the generations.
But that is good, no? Because whole evolution is driven by variability. So more and more variability you induce in a system, there is a chance that you will evolve into something bigger or better.
Something drastic.
Even drastic, what is the problem with that? Tomorrow it's like
Your system will be able to handle that.
So that evolution is
no, even directive, it doesn't matter that whether it's a directive or not. But the important thing is like if a system can create variability at a faster rate, like in case of viruses and all, it's advantageous.
Your system will
Or
not
the
be able to
bacteria.
handle that. Your system will not be able to handle that. I got deleted.
no, we are not fighting for survival.
Who told you? We are not now, because we built house, we built medical system, and we built PGI, so we are not fighting for survival. But still the animal in the forest, they are still fighting for survival every single day. A tree standing in the rain, it is fighting for survival every day. It's fighting against insect, because tree cannot spray insecticide themselves.
Is it because even from the most primitive organism to the most evolved ones, so-called evolved ones, the basic mechanism of genes, transcription is almost similar?
So yeah, I mean that is the idea, but I will actually put it little more refined way. I understood what you are trying to say, and that is what the idea is. Anybody would like to attempt one more time?
But like in case of COVID, because we have generated vaccine, so another strain comes, so that can fast. So that means that virus was surviving for their survival. Fighting that.
Yeah, but that is advantageous. No, that is advantageous. No, no, but in normal atmosphere also, when you don't have vaccine, virus actually mutate fast, very fast. They actually evolve. The new strains comes. And bacteria does that all the time. Many organism does that all the time. So there is nothing wrong being fast. Okay, unless you are not driving in Chandigarh roads, you can be fast. Okay, still fast. But the issue, it's not about the fast. So what is really happening there? That the idea. No. So
Take the survivor itself.
the explanation is more simpler. Okay, this explanation is way more, and actually that is
So during cell division, the chromosomes they are derived from the previous cells, so they are conserved in that manner that they will not change drastically because one set of chromosome comes from.
no, but that is also not true. See, you think about cancer. How drastic changes has happened in the cell, and everything changes, like the cells don't look even anything like what the tissue of origin is.
That drastic change, that is not surviving.
They are surviving. Cancers are better survivor than. No, the cancers are better survival than the human being who is carrying those cancers. They actually relapse, they metastasize, everything they do, they play havoc on the system. And even body's own immune system cannot do anything about it.
The basic mechanism of life is so worked out in the primitive to the that if I don't want to mess up.
No, why that is worked out? That's the question.
If we genes aren't retained, then they basically become more mutable, right? And at that point, you lose out on basically you don't have the chance to prove that this trait will survive.
No. Okay. Let me give you a clue. Okay. So whatever be the answer of this question is, it has to do with this class. if actually you put your answer in perspective of what I'm going to teach or what I was teaching for last 40 minutes.
It will all not be in harmony.
So the issue is that gene actually works in networks, and basic gene regulatory networks are conserved across species. So the same module of genes or set of genes are actually used again and again and again during evolution. Yeah, it's not that why do you want to change. The problem is that when so it is like you are 5 people are actually going for a hiking, and each of you are given a role. It's very hard for one person to deviate from that. Otherwise, you are very arrogant. You are full of yourself. Unless you are such a person, it's very unlikely that throughout the course of your hiking, you will deviate from that path, because a role is assigned to you. And it is not because it is because of that group dynamic. Group is actually has to behave according to the roles assigned, and that means you will less chance that you will veer. So that's why genes don't mutate in a evolutionary time, and that is the first thing. Second thing, the same pathways are reused in like heart development. If you look at vertebrate heart development, the same basic central network is actually, the FOXP network is actually used again and again and again. Similarly, if you look at body forms like bilateral symmetry generation or neuronal symmetry like anterior posterior axis generation in any organism, bilateral organism, it's always Hox genes, and how they pattern Hox genes based on anterior posterior polarity. So the same network, same set of genes are reused again and again and again across the, and this is only possible because genes works in networks. They don't work alone, and lone wolf has no role in biology. Okay. Okay, so let's start. You have a Jupyter notebook. This is called as function analysis, and you have a file which is this one. You can just open and see what it has. I just actually made a small file for you. It has gene ID. So this has gene ID. It has base mean value. I'm not going to use it, but just it is there. Log2 fold change, which I'm going to use, p value, p adjusted value, and then I think it has other 2 columns, which is two more IDs. One is entrace ID, another is ensemble ID. So I have this. Okay. So I have 3 kinds of ID in this table. I have log2 fold change, I have p value adjusted value, and so this is a table. This is the classic table that you get from the differential gene expression data. And unless if you don't have any of these gene values like any kind of symbols, entrace value or ensemble values, I will actually, if I have time, I will show you how to get those values. Otherwise, there are other easier way to do it. But let's believe that you have all these things. Okay. This is a notebook. Essentially, you don't have to load this library, but for some reason, I am actually not able to get it automatically loaded. I don't know why. So what you do, just go to this column, cell, and run that cell like as you do, shift enter, and it will load you all the packages that we need. Okay, just do a shift enter or you do run, and you will be able to do it. So it just loaded all your packages, and we need few packages like cluster profiler, we need path view, we need SPIA. We are using human samples. So this database is HS. If it is mouse you are using, then you use M capital and small m, or if you are using Drosophila, use big D and small m. Okay, and then other databases are there, but these are the major common organisms that people use. Now, so this is running, all thing done. So now we go to our file. So this is familiar to you, we have used it many times. So these are a CSV file, and we are going to read this CSV file into an object called as gene list. So we will do that, and so we did that. I am going to do something. I am just going to add one cell here, and I want you to do so and just check this file. Okay, so how you will check? You will do a head and you will say gene list. Okay, and I will see, okay, the data looks like this to me. This is the first five lines of this data. There are some gene names are there, and all this different columns that I need is there. I can also check what is the dimension of this file. So I will use dim, and if I do a gene list, then so it has 25,665 genes or rows, which is actually each gene is probably for a gene, and then you have 7 columns. Okay, so just we are exploring our data set. Now, next thing we need to do. So next thing we need to do is to subset this this data set. So as I mentioned, like all this Aura kind of like enrichment analysis platform, the biggest challenge is that we need to take responsibility. So as Spider-Man said, with great power comes great responsibility. So we also have a great responsibility, and one of the great responsibility we have is to subset and find the right criteria for subsetting, and there is no criteria such as right criteria. I am just using a criteria here. I am just saying if the absolute log 2 fold change value is twofold, that means twofold up regulated or twofold down regulated, then yeah, okay.
Click on the folder and close that section completely.
Okay. Okay. So I'm using a command called as subset. This is a base R command. You can actually use what Francis told you in the dplyr also, but then here actually I'm using a subset command, and what I want to subset, I want to subset my object called as gene list. So this is there, and based on which column, I am based on the log 2 fold change column, but I just want absolute value. The reason why some will be positive direction, some will be negative direction because of up regulation and down regulation. I don't want to actually distinguish between them. So anything that is going either twofold up or down, just subset them and give me that and store that in a new object called as gene. Okay, so I am saving that into a new object called as gene. So now I have two objects. So are you able to run this? So if you look at dimensions of gene object, you will see there are 509 genes. So basically I have 509 genes which is twofold up regulated or down regulated in my list. I'm just kind of not using p-value here, but then you can also have multiple criteria, p-value and others. Okay, now let's actually, and in this notebook I have added some notes also for you to read, that what it does, what this terminologies means and everything, so you can always read them. Okay, now we want to do a Go term analysis. So as I mentioned to you, that there can be 3 different ways of doing Go term analysis, or 3 different hierarchies are there. One is biological processes, molecular function and cellular component. So the first thing what I am going to do, I am going to do molecular biological process, and I want you all to do molecular function and or one of them, CC or MF, whichever you want to choose, you do that. Okay. So what are the terminologies, that what are the information that we need? We need here, this is the function, this is a function from cluster profiler. Okay, enrich Go, this comes from cluster profiler. We have subsetted our object and we saved them as a gene new object which has 509 differentially expressed gene, and then we want to take the gene list from ensemble column. So that is why we have a $ and ensemble. You can also use with the other IDs also, but then I am actually using this enrich Go, takes usually ensemble. Then you have to give what kind of organism you are working with. So in my case I am working with HS or human. If it is mouse, then that will be org.mm eg.db. So you will use organism database as the organism you are using. Now third one is what kind of key I am using. What is the key means here? Here actually the what kind of gene IDs I am using. In case of me, I am using ensemble ID. So I said like my ID is ensemble. If I am using symbol, then it will come, symbol will come. Ontology, what ontology term I want to, in this case I am doing BP, yeah, or biological process, but if you are doing molecular function or cellular component, you will use MF or CC, and both in capital, okay, no small letters there. Then what kind of adjustment I want to do? It's actually Benjamin-Hochberg correction, that is why BH is mentioned there, okay. And then there is a p value cutoff and q value cutoff, and forget about this part. Only thing you should remember that when you run this command, whatever organism you are using there, you should use the same organism database here, okay. And then if you run this command, it will take few minutes to complete. And it's doing all those Fisher's test and adjustment of p-value adjustments and everything. And if you want to run, you can also run molecular function and CC along with me. Make the changes and then just run them. Would you run? So Q value is actually equivalent to FDRs, or it's adjusted p value basically. Either way you can. People uses it. That is not the real. I mean, if you ask a statistics person, probably they will get offended. But for biologists, Q value can be FDR, adjusted p value equivalent to that. So for us it is fine.
So you told that we don't have any corrections, those corrections are harsh and we may lose out on the positives. FDR we are using. So why have we kept that?
Most of the statistics actually based on FDR is a relatively new way of doing it. So many packages are actually still using all Bioinformatics or Benjamin-Hochberg corrections. Okay. So my run is over. Okay. Now I actually want to plot them, and so before that, what we'll do, I'm done with this. So okay, here if I go here in the dot plot section, and I will add a cell, okay, and then I will ask head, and I will say ego2, because this is the object that we created. You see this one, top we have created that object as an output for my ego2. That was my output for my go term analysis. So I want to see that if I found any terms in that, or what are those terms, okay. So I just want to see a head of this, and it will give you, and don't worry about much of these things. So it actually tells you few things here, that what are the go term ID and gene ratio, which is basically fold change in this case. Then you have p adjusted value, q value, and this gives you a list of genes which are these are actually genes here, but these are RSV data. But forget about the biology of it at this point. We are just looking at how they. So it's a big table, and you can write it out, like you can write as an Excel sheet, and you can use that for, like you can handle that as an Excel sheet also, okay. But we are not actually interested in that. What we are interested in, can we actually make some nice plot out of this? And one of the beautiful plots you can make is a dot plot, and that is pretty simple. See, with just single command, dot plot, and within the parenthesis ego2. Ego2 is the object that we created from our go term analysis, and it gave you a beautiful dot plot with all the enriched terms here. Then p value is actually colored based on the based on this matrix. Like lower the p value, it is more red. Higher the p value, it is actually blue. And then the size of these dots are actually according to the count, how many genes are there represented in your data. That is how, if more genes are there, the dots will be bigger.
On the x-axis, it is written as gene ratio.
it's a fold enrichment basically. Gene ratio is essentially the fold enrichment analysis in this. Then same thing you can actually also plot as a bar plot. Okay. So say it's the same data. Now actually you are being shown as a bar plot. So instead of dot plot, I like personally dot plots more because it looks a little more cleaner. But bar plots, a lot of people like to actually put them as a bar plot too. So this is a bar plot. Again, in this bar plot, they are on an x-axis. They are putting count. So basically, this is the number of genes in your differentially expressed table with that terminology. Okay. Then this is another way of making bar plot. It just showing only the 12 categories, but you can restrict anything. But the main thing I want to actually, these are like simple plots, dot plots, and those kind of thing. But I want to actually show you two plots, and that is the key plots in the analysis of gene regulatory networks or this kind of analysis. So one is called as EMAP plot. Okay. And you have seen these plots actually in a different context, but this one is called as EMAP plot. What I will do, I will actually plot them as PDF. I think that is better, and I will save them as PDF because there is a why I will show you why I will do that. So let me actually explain you this command. So you have seen Francis has used this many times. So PDF, and then you want to save this in this name called as EMA ego2.pdf. So this is the name I want to save, and I am putting a width and height argument. And the reason why, these are big pictures, and if I actually use a default which is 800 by 800, then I won't be able to see it. So I am just doing that. So, and then this is a command I want to use, emap plot ego for the ego2, and then this I'm actually closing that plotting engine with the def.off option. So just let's run this, and I believe I have the plot. Where is the plot? Yeah. So, okay. Oh, this is okay. Let me actually do on something else. This is it's saying like dimension is too big. I will go for a PNG and then. PNG. Okay, so this is your EMAP plot, okay, and do you know what we are seeing here? Networking. No, not networking. What are we seeing here? Nodes of what? So no, so in this actually they are not genes, okay. So what we are seeing is that how, suppose you have 10 or 15 pathways got enriched. Not genes. Here it's not talking about genes. So what EMAP plot shows you that you have 10 or 15 pathways that you saw the enrichment. Now the question you are asking, are these pathways are related to each other? Basically, like you are looking for a bigger, what is their relationship with each other? In this, all the pathways are related to each other, but there will. Red dot is a pathway. Each red is a pathway, but you know there are instances when all of them will be separate. Like you will be having like 3 or 4 pathway together, two will be there together, one will be like lying out. So you know that okay, there is a module. The modules of pathways are also, and they are how they are interacting with each other. But I will actually go to next plot. Then we will actually come back to this again.
Can you show the dimension for this? Change dimension.
It's 200 by 200, 2000 by 2000, and because it's a bigger plot, that's why I wanted to.
2020.
Okay. Sorry, I'm actually going a little fast, but my okay. Now the next plot that we are working on, the next plot that we are plotting on this same data is called as CNET plot. Okay, and CNET plot is what actually Dr. Amit was just mentioning. It actually shows you the relationship between genes. So you got all these pathways. Okay, now you see. So this is if you look at, it is showing you that how genes are actually related to this pathway and if a particular gene is shared between multiple pathways or not. Okay, so this is what actually you can do this kind of analysis, this kind of plots you can plot and do the analysis for your. Now,
can you show the command for this? CNET?
Yeah, it's just same. Yeah, sorry. हाँ वो हाँ जस्ट उसको खाली रन कर दो नीचे आ जाएगा वो ओके anyway so for most of this kind of analysis you need this four kind of plots. You need to have a dot plot or a bar plot, and then you also need a CNET plot and EMA plot to see how genes are related to each other or how. And I will actually show you one other example because I was actually doing one analysis here. This is my because that has little something different. Okay, here you are. So, can you see this? So, this is a different data set. Okay, I thought I should have used it. So, now you see here, you have this pathway. Here you have a second pathway, you have a third pathway. Now, between these two pathways, these genes are shared. These genes are unique for this particular pathway. Similarly, these genes are unique for this pathway, but these genes are shared between these two pathways. So, the enriched pathways what you got and the genes underlying those pathways, how they are shared, you are able to figure out, you are able to find that using your CNET plot analysis. So, this is a different data set I actually I had. And similarly, I am going to actually show you a EMA plot also, EMAP plot also on the same data set. And then you will realize that what I am trying to, okay, I don't know whether. Map or PNG. So here is the EMAP plot, okay, for that data set. Now you see, all of these pathways are actually interrelated with each other. Okay, this is one set of pathway, they have some kind of a relationship. Now you have another set of pathway, they are interrelated with each other. You have a third interrelated pathway, but while there are 3 or 4 different pathways that are just, in most of the cases, these kind of pathways are, even if they are enriched, they may not have much of a significance. Because if these kind of pathways, there are possibility that there are multiple pathways coming as an enriched pathway, but they are part of a module, then likely that they might be more significant, or the one something that you need to pursue in your experimental data. So EMAP and CNET plot, you saw like both of those plots are very useful. And why you do EMAP, CNET plot, one thing is looking at the interaction, but what are the other reason why you do use it?
It is picking up the one particular gene where the
Yeah, basically the thing is, sometimes what happens, only one set of genes will be driving everything. You have 10 pathways, but all the 10 pathways have the same gene. That means only one of those pathways are right, not anything else. So CNET plot will also give you what kind of diversity is there in the gene usage when they are doing this kind of enrichment analysis. Okay, so is this useful? Say something. Yes? Okay. Now you are looking at Go term. Many of you might have done molecular function or CC. So basically you can, just by changing one small command ontology, you can identify all the 3, and whatever output you are setting, you can actually run them through all the 4 plots and you can generate your plot. Oh, it's R. It's R actually. You are in hardcore R, you are doing coding. Okay, and you can actually, what I was using just now, I showed you, that was RStudio. So you can run all this packed thing in the RStudio, and I will send you a RMD file for that too. I will tell you what to do. Okay. Now KEGG pathway. So similarly like Go term, you can do a KEGG pathway analysis. Okay, and then so this is the code that you will use. Here everything is like clear. You are using entrust IDs for the genes. So same, the object gene you are using, organism what you are using. So in the KEGG pathway analysis, it uses terminologies HSA, MMU, MMU for mouse, HSA for human. So we are using HSA, and then we just gave a p-value cutoff which is 0.05. And once we are done with this, I think it is done. Yeah, it is done. And if I want to see if I have any term, then I say head and KK. That is actually our object, and it shows you that various pathways that are Go term. Now this is giving you the KEGG ID, KEGG pathway IDs, and then its enrichment values or p values are in. Okay, and then if you take that KK object and you can make dot plot, you can make bar plots, you can make EMAP plot, you can make CNET plot, and you can depict all of them. Okay, and then we will come back to this when we are going to do a path view analysis. Okay. Okay. Now, so I am not going to do the plotting. Now, you remember, like I was talking to you about the problem with Go term analysis. We have to take larger responsibility of setting up a threshold. But many of us, I think we should not take that responsibility unnecessary. So GSE analysis actually uses, take that burden off from us. So if you have a differential expression gene list, you don't have to make any cutoff. Rather, you give whole gene list, and then you tell program to actually analyze the data based on, even small changes will be taken into account if it is a part of a network. And that is what we are doing here. The first part of this analysis, what you are doing essentially is to just clean up your data. You are just cleaning up your data. So you are taking the gene list, which is the all genes list, and then you are taking an entrust ID, and you are removing any row which is NA. If the entrust ID in a row is NA, which is not there, then you want to remove that row from your analysis. Okay. The second line, you are actually removing any duplicate row. If you have a row where the entrust ID is duplicated, like both the rows of same entrust ID, it will actually remove the second row. The first row it will keep and it will remove the second row. So you are first line of this code, you are removing NA. The second line of code, you are removing the duplicate, and then you are passing off the log2 fold change value into a new list. Just it one log2 fold change value, and it is leaving that to a list called as GSEA list, and you are adding the name of each of this one from the entrance ID. So this is, you actually created a list called as GSEA list, which has no duplicate entrance ID. It has no NA in the entrance ID column, and you have a clean. NA usually what happen, yeah, no, there won't be any gene name. So what happen, programs usually put NA there. So they don't want any row which has, no, there's no ID. That is why NA is there.
But that number that
the symbol might be there, and sample ID may be there.
symbol is not there, but that ID is there.
Which ID?
So that ID gene has not been identified. That I mean,
Maybe, yeah, maybe. No, other way around also. Suppose you have a gene symbol. Say I discovered a gene called in my name, like Boni De Kumar. I gave a gene ID like gene symbol as Boni, BONY, and so ensemble also I got, but entrance there is no ID. So that means when I am actually annotating that gene for entrance ID, it will say NA.
I'm saying that entrance ID is giving a number.
Yeah, it will give a number, but instead of that number, there will be NA. If that number is not assigned to that gene.
Three ways of depicting a gene.
Yeah.
Ensemble, entrance, or that whatever.
Yeah.
Gene ID. So we are picking up only this. If
This
there
one.
is NA,
Then this NA we want to remove it, and if there is a duplicate also, then we also want to remove it. Okay. So just run this line of code and you are able to generate a gene list. Okay. We created the gene list, and then what we are doing, we are actually doing same thing what we did with Go term. So only difference is that the gene list what we created up, we are taking that and running into and a new function we are using called as GSE Go, and then we are creating and then we are using this object. We are using organism, and same like we can use biological processes, molecular function, or cellular component, whatever you want, and then just run this. Okay. It actually also do some permutation, so that is why it is asking for nperm, how many number of permutation you need to do. So do all those kind of stuff.
I forgot इसमें और पहले वाले में क्या था?
बस पहले वाले में हम कट ऑफ करके उसको लिस्ट देते थे, और यहाँ जो है वो हम कट ऑफ सारे ले रहेगा, उसके हिसाब से वो करेगा वो, ठीक है. So let's run this and yeah. So same like Go term, and then we are going to make all the plots, whatever Go term plots we made. Will you guys be able to do it yourself? No, I'm saying like after this. See, the thing is, you can run same set of code. Okay. Only thing what you need to do is to change the first first object and create the object. And if your header names, like the column names are different, just change the column names and just run it, and it will create output for you. Okay. So you don't have to really rewrite all of this code for your rest of the analysis. So I'm going to see if I got anything. So head ego 3. So see, I got some terminologies. Here with the IDs and enrichment values and those kind of stuff. So you can do this for biological processes. You can do this for but you will get a different terminologies compared to. Okay. Now. Okay. Now the question is, I want to show everything together. Like if I have a pathway and I want to show like what genes are actually changing and in which direction they are changing. I want one single picture for my presentation or for my paper. So suppose if I am actually interested in this pathway called as influenza pathway, and I want to see like if influenza pathway, what are the genes that are changing in the influenza pathway, which direction they are changing. So I can use a package called as path view. Okay, and so this part is actually same thing what we did, that we actually removed any NA, we removed any duplicate, we subset our actually gene list at a p-value cutoff of 0.05, and then we are running a path view on this. So basically this is the same thing that we were doing, and I will show.
Whole data?
No, so whole data you should take. Yeah, but what we are going to see is only one pathway now. Okay, we are. I will actually show you path view. So in this one, so we created this list here, and that list we are. Oh, sorry. So yeah. Okay. So in each pathway, you have a ID like this, and where you will get this ID from. So if you go back into your Keck pathway analysis here, and if you look at head, you will see like there is a pathway ID is there. So if you want to see any of this pathway ID, you can use. We'll use one of this. Okay, let's use that.
It could be Go. It could be.
It's basically it's basically Keck pathway you have to use. Okay. So suppose I'm going to redo this analysis with this. The one pathway which was enriched in our data set. So the only thing that you need to change, oh, what happened? But anyway, let me show you. After that, we'll come back. So this actually put your all the genes in that. So what it did, yeah, I will just finish with the path view and then we'll come back. So what it did, it actually took your genes from the differential gene expression did, and it laid it over the pathway. And what you are seeing here, that in this case, fortunately or unfortunately, all the genes are upregulated. But if there is a downregulated gene, then you will get a green color there. So in one go, you can depict your whole enriched pathway or enriched pathway into single thing. And if you have 4 or 5 different pathways of your interest, plot each pathway individually. And how you do that, you will just change. Oh, where is it? Oh, where is this? So yeah, just change this pathway ID. Okay, here this pathway ID you change, and you will get the new pathway. And there's one thing. This is the only package I know where you don't have to use that PDF or that the storing. It will directly store into your whatever working directory it is there, and it will show you that. So just change this. If it is a different organism, change the species also. If it is mouse, then make it MMU. If it is Drosophila, make it DMA. Otherwise, human, keep as such like HSA. Okay. So we have done enrichment analysis using 3 different ways. We used GOTERM analysis. We did GSEA. We also did Keck pathway analysis. We learned 4 different ways of plotting them: dot plot, bar plots, EMAP plot, and CNET plot. And we learned how to interpret EMAP plot and CNET plot. And we also learned how to plot your Keck pathway data into path view. And then, so I think with this, if you can do this much, you are very comfortably, you can write a good paper also and use some of this. So these are like publication quality figures, so you can actually use directly for your publication or for. So I think we should go for a T, and we will actually come back, and then Manpreet will actually quickly show you that all these things what I told you, you don't need to know. She will be actually show you a more clever way to do this.
One more thing. So we classify or we cell type clusters using calophysics manually.
Oh. So, hi everyone. Hello, hello. Hello. Let's start because we have a talk after this. Can we start? Yeah. Okay. So I'm gonna help you in a way because all that you've been doing for the past 2-3 hours, I'm gonna show you a very simple way to do it, and you're gonna ask Dr. Boni why he made you do all that, right? So there are some web-based tools which you can use in place of going through all the or jumping through all the hoops. You can just simply use a web-based tool. As he also mentioned before me, there's David, there's Panther, ShinyGo. There are a lot many web-based, but you have to be sure that the database or the tool that you use, it needs to be updated, right? It cannot be like which was updated 5 years ago. So be careful of that. So for precisely that reason, we recommend that you go for David. Okay. So what does David stand for? I'm really bad with full forms, right? So basically you can just Google David and write, yeah. So this is a database for annotation, visualization and integrated discovery. That is what you were doing, right? So simple GUI web-based tool which you can use, right? So so I'll just quickly show you how you can do very simple analysis here. If you remember, I mailed you some data sets day before yesterday. So I'll just use that because you already have it on your system. Yeah. So I think all of you have downloaded this. So this was one of the files. These are the filtered DEGs, right? You can see from the file name. So from this, you can just simply copy the gene IDs because they're filtered DEGs, right? So copy this and paste it to your tool. Very simple. Start analysis. So upload. You can paste a list or you can upload a list, anything, right? So from here, identifier. What have you uploaded? What did I upload? What? Yeah. So this is the ensemble gene list. Yeah. So you can put that or even not. It's okay. So you put it here. And then, okay, so you have to be careful here. This is a gene list, not the background. So background would be the list of like a universal set. So your DEGs are a subset of the universal set, right? You know that now, right? They're not all the genes that you're working with. It's only a significant subset of genes. So you have to be sure that you mention that this is a gene list and not the background. You can upload that as well. And finally, you just have to submit. And that's it. And now you can start looking at the results. So no need to do R or things like that. Yes, so he's saying that I should have told you this earlier, and that is why we did not tell you this earlier, right? So we have a weird pleasure in troubling everyone. So once you have this, so now you can start analyzing. So you can read this. What do you think? Functional annotation, right? This is what you did. So you have function. So I'll just show you quickly what it looks like. Let's just start with the functional annotation chart. Okay. So now you have all these summary details, right? So this, it can automatically realize that it's from the mouse. These are not human genes. It's from the gene list that you were working with yesterday. So once you have this, so you can just by using the defaults, you can actually check for the, click for the chart. What happened? No, I think my net died. Is your net working?
No.
Okay. Let me just see. Something's wrong with my system. I don't know. Oh, it's coming. It's coming. It's fine. Right. So can you see? You have a small list. My screen is small. So now it's telling you your geo terms, right? And the different, so I'll just quickly run. So this is like the new version. So a disclaimer, I haven't used this tool in the past 3 years. So it's been a while, so I'm also figuring it out. But so I would like to switch to the classic view, but the net is not working.
So you can go into it.
There's something wrong with the net. It's not stable. I don't know. I don't know. There's something wrong with the net. Yeah. No, there's no firewall. This is a.
जीन ontology में चले जाओ.
No, it's nothing to do with that. वो कुछ background में problem है. Okay, okay, fine. Let's just. Maybe I'll have to upload it again. कुछ नेट का इशू आ रहा है. I don't know. Yeah, I'm sorry. The net is refusing to cooperate. I don't know what to do. Or maybe you can do it after the Zoom call then for 5 minutes if you're interested. No, but there's something wrong with the server. It's not working. हाँ जी बोलो. हाँ? Sorry. We can share it. Oh, sorry. Maybe. I think Dr. Boni said it's not working. I think we'll have to do it later. नहीं हो रहा है. It's. I think there's something wrong with the David server. चलता है फिर बंद हो जाता है. It's coming and going. We'll try uploading it once again. नहीं वो बंद हुई जा रहा है बारी बारी.
चार्ट की जगह पर यहाँ वाला जो आता है उसको ले.
वही किया था पहले वैसे. Okay. Okay. So I'll just probably not. I'll just simply show you the clustering first. फिर से ना बंद हो जाए. Let's just start now. Yeah, there's some internal server issue. We can do it later. Yeah. Something is wrong. Yeah. So anyways, but the reason I was just showing you this, that if you feel overwhelmed when you start working and you want to visualize the data, this is a good tool. But there's a problem. There's a limit to the number of genes that you can upload, as it is with any GUI based thing. So that is why you will eventually need it. But for your basic analysis, you can start working this. And probably when it works again, I can just show you what it looks like.
We should. Our way is because will give you more flexibility of doing it. Data will be under your control. You can change things. You can change how it looks.